Good morning, my dear students. In this session, we are going to discuss about introduction to entomology. Entomology, it is a Greek word. Entomon mean insect, logos mean study. That is, scientific study of the insect is the entomology. The term insect comes from the Latin word insectum. It means the insect body is divided into three sections, that is head, thorax and abdomen. Then what do you mean by insect? See here, any organism in which body is divided into head, thorax and abdomen possesses two pairs of wings and three pairs of legs. Such organism is known as insect. Then what do you mean by pest? Any organism which is noxious, destructive or troublesome to man or his interest is the pest. Examples are insects, rat, mites, weeds, etc. On this earth, man originated about a million years ago, but insects came into the existence 350 to 500 million years ago. Arthropods are originated during Cambrian period of Paleozoic era. Insects are originated during Devonian period of Paleozoic era. The earliest known fossil insect is Raniella precursor. It is a columbolan insect from the Devonian period. Insects belong to kingdom Animalia, phylum Arthropoda, and class Insecta. And class Insecta is the largest of the whole living organism, and it constitutes 72% of all living animals. Thank you. In the next session, we are going to discuss about body organization in insects. Good morning, my dear students. In this session, we are going to discuss about insect body organization. Insect body organization refers to the structural arrangement of an insect's body parts. See here body segmentation in insect. Insect body generally contain 20 segments. That is, head contain 6 segments, thorax contain 3 segments, and abdomen contains 11 segments. Insect body is differentiated into 3 regions, that is head region, thoracic region and abdominal region. Each region is also known as tagmata, that is head tagmata, thoracic tagmata and abdominal tagmata. Each tagmata is composed of group of segment, that is 6 segment in the head, 3 segments in the thorax and 9 to 11 segment in the abdomen. That is, grouping a body segment into a distinct region is known as tagmosis. The arrangement of the body segment in the embryonic stage is known as primary segmentation, while in adult insect is known as secondary segmentation. See here the structure of insect head. Insect head, it is the anterior most stigmata of the insect body. Head capsule is sclerotized and articulated with the appendages antenna, such as antenna, eyes, and mouth parts. Head capsule without appendages and with only sclerite is known as cranium. Head capsule, it is made up of 6 segments, that is 1 ocular segment, antennal segment, intercalary segment, mandibular segment, maxillary segment, and labial segment, and provided with the following appendages, that is eyes, antenna, and mouth parts. Then what is the function of the head in insect? Head, it is mainly concerned with the feeding and sensory perception. See here the sclerites and suture of insect head. In the insect head or insect head capsule is formed by the union of several number of cuticular plates, that is labrum, clypeus, frans. Frans is the largest sclerite of the insect head, next vertex, next ocular sclerite, antennal sclerite, acciput, and post-acciput. All these sclerites are commonly form a head capsule. That is, head capsule is formed by the union of number of cuticular plates are known as sclerites. These sclerites are joined together by means of cuticular line or ridges are known as suture. See here, suture of insect head. The cuticular plates are joined together. The cuticular plates are joined together by means of cuticular lines or ridges are known as the suture. See here the different suture. This is the clypeolabral suture, next epistomal suture, next antennal suture, genal suture, ocular suture, and epicranial suture. Epicranial suture is also known as line of weakness or ecdysial suture because the exuvial membrane split along this suture during the process of molting or ecdysis. See here, this is the post-occipital suture, and this is the only real suture of insect head. It's so called because it separates insect head from the neck. See here the types of insect head. Based on the bending or inclination of long axis of the head and orientation of the mouth parts, there are 3 different types of insect heads. That is hypognathous head, prognathous head, and opistognathous head. See first about hypognathous head. Hypo means below, gnatha means jaw. Here the mouth parts are directed in downward direction. This is also known as orthopterate type of head. Here the long axis of the head is vertical. Long axis of the head is vertical and it is at right angle to the long axis of the body. In this case, the mouth parts are ventrally placed and projected downward. This type of head is found in grasshopper, locust, and cockroach. Next, prognathous head. Pro means front, gnatha means jaw. Here, the mouth parts are directed in forward direction. This is also known as coleopterate type of head. Here the long axis of the head is horizontal and in line with the long axis of the body. Mouth parts are directed in forward direction, and this type of head is found in beetles and weevils. Next, Opisthognathus head. Opistho means behind or back. Gnatha means jaw. Here, the mouth parts are directed in back side direction or in backward direction. This is also known as hemipterate type or opisthodorhynchus type of head. Here, head is deflexed and mouth parts are directed backward and held in between the forelegs. See, the mouth parts are directed in the backward direction, present in between the forelegs. This type of head is found in stink bug and red cotton bugs. That's why this type of head are known as hemipterate type of head or opisthodorhynchus type of head. Next, what do you mean by procephalon and gnathocephalon? See here oral cavity. Preoral region is known as procephalon. Post-oral region is known as gnathocephalon. Thank you. In the next session, we will discuss about insect neck, thorax and its appendages, abdomen and its appendages.
Good morning, my dear friends. In this session, we are going to discuss about insect neck or cervix, thorax and its appendages, abdomen and its appendages. Let's see about neck or cervix. In insect, head is connected to the thorax by means of a membranous region. See, this is the membranous region is known as neck or cervix. In insect, head is connected to the thorax by means of a membranous region is known as neck or cervix. This membranous region is quite flexible and allows the free movement of the insect head. Let's see about thorax. It is the insect head and it is the insect abdomen. The region which is present in between the insect head and abdomen is known as thoracic region. Insect thorax is the second or middle tagmata and highly musculated due to the presence of bases of wings and legs in them. The three segments of insect thorax are: first segment is prothorax, second segment is mesothorax, and third segment is metathorax. See here also, the first segment is prothorax, second segment is mesothorax, and the last segment is metathorax. Each thoracic segment contains a dorsal scleroid is known as tergum or notum. Lateral scleroid is known as pleuron and ventral scleroid is known as sternum. See here also, dorsal scleroid is known as notum or tergum. Lateral scleroid is the pleuron and the ventral scleroid is the sternum. See the appendages associated with the thorax. Insect contains two pairs of wings. First pair, that is forewing, second pair, that is hand wing, on meso and metathorax. Due to the presence of wings on meso and metathorax, these thoracic segments are commonly known as pterothoracic segments. Insects also contain three pairs of legs on each thoracic segment, that is on prothorax, foreleg, on mesothorax, midleg, and on metathorax, hind leg. In addition to the wings and legs on thorax, two pairs of spiracles are also there on the pleuron of meso and metathorax. The spiracles are the organ of respiration through which air is entered into the insect body. Due to the presence of wings and three pairs of legs in insect in this region is mainly concerned with the locomotion. Let's see about abdomen. This is the abdominal region in insect. Abdomen is the third and posterior most stigmata of the insect body. It is made up of 9 to 11 segments. And this region is highly flexible, and this flexibility is most important and required for copulation and oviposition purpose. Each abdominal segment is made up of only two sclerae, that is the dorsal sclerae is the tergum or notum, and the ventral sclerae is the sternum. There is no pleuron in the abdominal region of the insect. Hence, the spiracles are located on dorsolaterally extended portion of the tergum. This is the extended portion of the tergum. On this, the spiracles are locating. In grasshopper, 8 pairs of spiracles are present in the first 8 abdominal segment, in addition to a pair of tympanum in the first abdominal segment. See, this is the tympanum present on the first abdominal segment. This is the auditory organ, helps in hearing in insects. Now see about abdominal appendages. Abdominal appendages in adult insects are genital organs and cerci. In male insect, the genital organs are located on ninth abdominal segment. In female insect, the genital organs are located on 8th and ninth abdominal segment. See, this is the cerci. A pair of cerci is associated with the 11th abdominal segment, and it is concerned with the sensory sensation. It is sensory. Cerci is sensory in function. Due to the presence of genital organs on the abdomen and a different system inside the abdomen, this region is mainly concerned with the reproduction and metabolism. See here the abdominal spiracles in catchidid or grasshopper. These are the spiracles. Organs of respiration through which the air is entered into the insect body. Now we are going to see female genital structure clearly. Female genital structures are located on the 8th and ninth abdominal segment of insect on the ventral side. The female genital structure or ovipositor contains three valves or valvulae, three pairs of valves or valvulae. See here, this is the first pair of valve, this is the second pair of valve, and this is the third pair of valve. Actually, from base of the 8th segment, valve for 1 will be originated, and this valve for 1 gives a pair of valvulae. First pair of valvulae. Similarly, from the base of the ninth abdominal segment, valvi for 2 will be generated and it gives off a pair of each valvulae 2 and valvulae 3. See here also in vertical section, this is the first pair of valvulae, these are the second pair of valvulae and these are the third pair of valvulae. Now we are going to see male genital structure. It is known as adigus also. This is the adigus of click beetle. This is the click beetle. It is belong to the order Coleoptera and the family Elateridae. The whole copulatory organ of higher insects, generally known as adigus. This adigus helps in insemination. In addition to the insemination, it's also helping clasping and providing sensory stimulation to the female insect. This adigas contain a median tubular penis, and this penis contain internally a inner tube is known as endophallus. This endophallus is aorted during the insemination. The ejaculatory duct which opens either at the tip of the penis or endophallus and ejaculate or release sperm into them. Lateral side to the penis are a pair of lobes known as the paramus. These paramus help in clasping as well as providing sensory stimulation to the female insects. See here, this is the ninth abdominal segment of brachytile and their reproductive organs. Thank you. In the next session, we are going to discuss about abdominal appendages in wingless insects, immature winged insects, and adult insects.
Good morning, my dear students. In the previous lecture, we have discussed about insect body organization, that is, structural arrangement of an insect body parts. We have discussed that insect consisting of three regions, that is head region, thoracic region, and abdominal region. Head region is the six-segmented part and it consisting of compound eyes, simple eyes, antenna and mouth parts are as appendages. Thorax region is the middle region and it consisting of two pairs of wings in the mesothorax and metathorax, a pair in each segment, and three pairs of the legs in all the three thoracic segments. We have already discussed about the abdomen contains 9 to 11 segments. 9 to 11 segment. Now we are going to discuss about abdominal appendages in wingless insects, immature stages of winged insects, and adult insects. See here I have listed abdominal appendages in wingless insects. These appendages are generally pre-genital, mean they are present before the genital organ. See here the list of the abdominal appendages in wingless insect, that is style, colophore or ventral tube or glue peg, retinaculum or tenaculum or catch, furcula or furca. In the next category, you see abdominal appendages in immature stages of winged insect, that is, in the larval stages of winged insects. Abdominal appendages in immature stages of winged insect includes anal papillae, tracheal gills, dolichastus, prolegs. In the next category, we are going to discuss about abdominal appendages in winged adults includes cornicles, caudal breathing tube, cerci, median caudal filament, pygostyle, anal style, ovipositor, and male genital organ in male insect. So first we are going to discuss about here abdominal appendages in wingless insects. I already told you that these appendages, these attaching structures are pregenital. Pregenital mean they are present before the genital organ. These appendages include styli in silverfish, colophore or ventral tube or glue peg on first abdominal segment of columbola, retinaculum or tinaculum or catch on third abdominal segment of columbola, furcula or furca on the fourth abdominal segment of the columbola. Let's see one by one by seeing their images. Styli. See here, these are the styli. This is the another styli. Varying number of paired tubular structures. Varying number of paired tubular structures found on the ventral side of the abdomen of silverfish are known as styli. These are the paired tubular structures which are present on the ventral side of the abdomen of the silverfish. So, generally these are the reduced legs, reduced abdominal legs. See, these are the thoracic legs, 3 pairs, and these are the reduced abdominal legs, and they help in locomotion. Next, abdominal appendages is colophore. See here, red colored colophore or ventral tube or glue peg. This is the structure. This is the cylindrical structure present on the ventral side of the first abdominal segment of silverfish, and it is protruded out by the hydrostatic pressure of hemolymph, and it acts as organ of adhesion, that is attachment. It also helps in absorption of water from the substratum and also help in respiration. This is all about colophore. Colophore, it is located on the ventral side of the first abdominal segment. See, this is the first abdomen. These all are thoracic segments. This is the first abdominal segment. So, in the first abdominal segment, on the ventral side, a cylindrical tube-like structure is there. It is known as colophore or ventral tube or glue peg. Next, retinaculum or tinaculum or catch. See, this is the retinaculum or catch. It is present on the ventral side of the third abdominal segment. In second abdominal segment, there is no appendage. In third abdominal segment, on the ventral side, retinaculum or catch is present. What is the use of retinaculum or catch? It is actually useful to hold the springing organ. So this furcula or furca is a springing organ in springtail. So when it is not in the use, this retinaculum or tinaculum holds it. So it is useful to hold the springing organ when not in its use. Next, we are going to discuss about furcula or furca or springing organ. It is a Y-shaped organ. It is present on the ventral side of the fourth abdominal segment. It is present on the ventral side of the fourth abdominal segment when it is released from the catch. When it is not in the use, this is held by the retinaculum or tinaculum or catch we already discussed. But when it is released from the catch, it exerts a force against the substratum and help in propelling up insect or jumping up insect in the air. So that's why we are calling it as springing organ or jumping organ or furcula. Next, we are going to discuss about abdominal appendages in immature stages, that is larval stages of winged insects. This category includes anal papillae in mosquito larvae, tracheal gills in needs of odonata and Ephemeroptera, dolichostre in antlion grub, prolegs in caterpillars, loopers, and semiloopers. See anal papillae, a group of four papillae. See here, a group of four papillae is present surrounding to the anus. In case of mosquito larvae are known as anal papillae. And these papillae are concerned with the salt regulation. That's mean they maintain the ionic balance and facilitate other metabolic activity effectively. Next, tracheal gills. These gills are the outgrowth of the body wall which are richly supplied with the trachea, that is respiratory tubes. That's why we are calling them as tracheal gills. And these gills obtain the oxygen from the water in the aquatic insects, that is needs of mayfly and dragonfly. See, lateral gills. Lateral gills are present in the needs of mayfly. These are seven pair in number. Next, caudal gills we can see in the needs of dragonfly. These are the leaf-like structure present at the caudal end. Next, rectal gills are present in needs of dragonfly, and these gills are retained within the abdomen in pouch-like structures. That's why we are calling these gills are rectal gills. Next, dolichostre. This is the antlion grub, and this is the antlion adult. This is the antlion pit. So, dolichostres, these are the tube-like structure. These are the segmental tube-like structure found on the ventral side of the body in case of antlion grub. These are the segmental protuberance which are provided or fringed with the setae. So dolichostre present in antlion grubs. These are the segmental tube-like structure and which are fringed with the setae. Next, proleg. See, this is the caterpillar. This caterpillar consisting of three pairs of thoracic legs, and these legs are known as true legs. And it consisting of varying number of, that is 2 to 5 pairs of prolegs. 2 to 5 pairs of prolegs.
So these are the abdominal legs. We are calling these legs as prolegs or pseudo legs. Prolegs or pseudo legs. Thoracic legs are known as true legs. Abdominal legs are known as prolegs or pseudo legs. So these are present in the looper, semilooper, and the larvae of moth, butterfly, and sawfly. These are present. These prolegs or pseudolegs are generally present 2 to 5 pairs. These are the unsegmented, thick and fleshy structures. Unsegmented, thick and fleshy structures, and the tip of these prolegs are known as planta. These planta are provided with the heavily sclerotized. These planta are provided with the heavily sclerotized hook-like structure are known as crotchets, and these crotchets aids in crawling and clinging onto the surface of the plant. So these crotchets are absent in softfly larvae. That's why the softfly larvae fell down easily when we will touch them.
Now you see abdominal appendages in winged adults includes cornicals, caudal breathing tube, cerci, median caudal filament, pygostyle, anal style, ovipositor, and male genitalia or male genital organ. Let's see first about cornicals. See in this image, these are the tube-like structures. These short tube-like structures which projecting from the dorsal side of the fifth or sixth abdominal segment of the aphids are known as cornicals. They are also known as siphonculi. They secretes waxy fluid and it serves as for the protection against the predator. Next, caudal breathing tube. This is the caudal breathing tube. It is present in water scorpion. Here, two grooved filaments closely applied to each other and forming a tube-like structure that help in breathing. Here, the tube extend above the water surface and allow the insect to access the atmospheric air while the insects being submerged. Next, cerci. These are the most conspicuous appendages associated with the 11th abdominal segment. These structures are sensory in function and exhibit wide diversity and form. See here, sclerotized or forceps-like cerci is there in airwig. Long and many segmented cerci is there in mayfly. Long and unsegmented cerci is there in cricket. Short and many segmented cerci. Short and many segmented cerci are there in cockroach. Short and unsegmented cerci are there in grasshopper, and asymmetrical cerci is there in male embed. One cerci is longer and another cerci is shorter. Next, a median caudal filament. In mayfly, the epiproct, that is, the topmost portion of 11th abdominal segment, is elongated into a structures like a median caudal filament, and these structures play various roles such as sensory perception, maintaining the balance, and propulsion in water. Next, pygostyle. See, a pair of pygostyle or a pair of unsegmented cerci-like structures are found in the last abdominal segment of squalid wasp. This structure is thought to play a role in enhancing flight stability and efficiency of the squalid wasp. We can also see pygostyles in Hymenopteran wasps. Next, anal style, a pair of short unsegmented structures. See here, a pair of short unsegmented structures which are associated with the end of the abdomen of the cockroach, that is male cockroach, are known as anal styli. These anal styli are useful to hold the female cockroach during their copulation with the male. Ovipositor, the egg-laying organ found in the female insect, is known as ovipositor. This ovipositor is suited to lay eggs in precise microhabitat, and they also exhibit wide diversity and form. See here, long and sword-like ovipositor is there in Catchidid, long and in long-horned grasshopper also we can see a long and sword-like ovipositor. Needle-like ovipositor is there in cricket. Sting-like ovipositor is there in worker honeybee. Next, pseudo-ovipositor. It is so called pseudo-ovipositor because here an appendicular ovipositor is absent in case of fruit fly and houseflies. See here, in fruit flies, the elongated abdomen is terminate into a sharp pointed structure which, with which the fly pierces the rind of the fruit and lays eggs. In houseflies, the terminal abdominal segments are telescopic, and these telescopic segments are used or which aid in the oviposition. The ovipositor of housefly is called as pseudo-ovipositor or ovitubes or oviscapt. Next, male genitalia, external sexual organ of male insect. External sexual organ of male insect confined to the ventral side of the ninth abdominal segment are the male genitalia. But in case of damselfly, the copulatory, the functional copulatory male organ is present on the ventral side of the second abdominal segment. Next, see some modification. We can see a reduced number of abdominal segment in many insects. For example, in springtail we can see only 6 abdominal segments. In case of houseflies, 2 to 5 segments, that is 2 to 5 abdominal segments are clearly visible and 6 to 9 segments are telescoped within other. In ants, bees and wasps, the first abdominal segment which is fused with the metathorax is known as propodium. The second abdominal segment is known as petiole, and rest of the abdomen is known as gaster. Thank you.
Good morning, my dear friends. In this session, we are going to discuss about insect body wall. Insect body wall is the external covering of the body, which is also known as integument or exoskeleton, and which is ectodermal in origin. See the structure of insect body wall. Insect body wall consisting of outer non-cellular layer, that is the cuticle, middle cellular layer, that is the epidermis, and inner non-cellular layer is the basement membrane. See here in the picture, here I have drawn cross section of insect body wall. This is the outermost non-cellular layer, cuticle. Below the cuticle, this is green color one is the middle cellular layer, that is the epidermis. Below the epidermis, the purple color layer is the basement membrane. See here, same. It is the vertical section of the body wall. This is from here to here. This is the non-cellular cuticle. Below the cuticle, this is the cellular epidermis, and below the epidermis, there is a non-cellular basement membrane. So, body wall consisting of outer non-cellular cuticle, middle cellular epidermis, and inner non-cellular basement membrane. Now see composition of the cuticle. Cuticle is composed of chitin, arthropodin, sclerotin, and resin. Then what is the chitin? Chitin, it is the main constituent of the cuticle, which is nitrogenous polysaccharide and polymer of N-acetylglucosamine. Chitin, which is water insoluble but soluble in dilute acids, alkalis, and organic solvents. Then what is arthropodin? Arthropodin, it is an untanned protein which is water soluble. Then what is sclerotin? Sclerotin is a tanned protein which is water insoluble. Then what is resin? Resin, an elastic protein which is responsible for the flexibility of the wing sclerites. So cuticle is composed of chitin, arthropodin, sclerotin, and resin. I already told you that cuticle is composed of three sublayer. They are, this is the cuticle. It is composed of three layers, that is outer epicuticle, middle exocuticle, and inner endocuticle. See here also, from here to here, this is the cuticle. This cuticle is subdivided into three layers. They are topmost, this is dark brown line is the epicuticle, light brown line is the exocuticle, and this is colorless line is the endocuticle. So cuticle is composed of epicuticle, exocuticle, and endocuticle. Epicuticle is non-chitinous. There is no chitin in epicuticle. Exocuticle is composed of sclerotin and chitin. Endocuticle is lighter in color and composed of arthropodin and chitin. Exocuticle, this exocuticle and endocuticle. Exocuticle and endocuticle are combinedly known as procuticle. So now we are going to discuss all the three layers of the cuticles. They are epicuticle, exocuticle, and endocuticle. We are going to discuss first about epicuticle. Epicuticle, this is the outermost layer. Epicuticle, see this is dark brown line. Epicuticle, this is the outermost layer of the cuticle and which is very thin. Pore canal present in exocuticle secrete or deposit the epicuticle. This epicutical layer is again differentiated into following five layers. They are The first and topmost layer is the cement layer, second wax layer, third cuticle layer, fourth outer epicuticle, and fifth inner epicuticle. What is the function of cement layer? Cement layer, see here, it is formed by lipid and tanned protein, and it protects the wax layer which is present just below to it. Then what is the role of wax layer? Wax layer, it contains closely packed wax molecule. See, these are the wax molecules and which prevent the desiccation, which prevent the insect from desiccation. It avoids the water loss from the insect body. Next, third one is the cuticle layer. Cuticle layer is the non-chitinous polymerized lipoprotein layer. Fourth one is the outer epicuticle. It makes the contact with the cuticle layer. Fifth one is the inner epicuticle. It contains wax filament. See, this is the epicuticle. It's again differentiated into five layers. They are cement layer, wax layer, cuticle layer, outer epicuticle, and inner epicuticle. So now we have completed about epicuticle. Now we are going to discuss about exocuticle. Exocuticle, it is the middle layer of the cuticle that is present in between the epicuticle and endocuticle, which is much thicker and composed of chitin and sclerotin. This exocuticle layer is dark in color and very rigid. Next, we are going to discuss about this endocuticle layer of the cuticle. Endocuticle, this is the innermost layer of the cuticle. This layer is made up of chitin and arthropodin. This layer is colorless, soft, and flexible. I already told you that exocuticle and endocuticle is commonly known as procuticle. So, in this slide, we have discussed about cuticular part, that is about epicuticle, exocuticle, and endocuticle. Below the cuticle, see here, epidermal layer is there. In the next slide, we are going to discuss about epidermis. See, we are going to discuss about this layer now. Epidermis or hypodermis. It is called hypodermis because it present just below the cuticular layer. Hypo mean below. It present below the cuticular layer. Then that's why we calling it as hypodermis or epidermis. It is present just above the basement membrane. That's why we calling it as epidermis. Epidermis, it is a single cellular layer which present in between the cuticle and basement membrane. The adjacent cells of the epidermis are held together by cytoplasmic process. See the cytoplasmic process. The adjacent cells of the epidermis are held together by cytoplasmic process known as desmosomes. All the epidermal cells are glandular and secrete cuticle and different enzymes. The epidermal cells are differentiated into following type based on the functions they perform. For suppose, consider this is dermal gland. This dermal gland produce cement layer, trichogen cell. This is the trichogen cell. Trichogen cells produce hair-like setae. See here, it is producing hair-like setae or trichomes. For suppose consider this is the molting gland which secrete molting fluid. For suppose it is consider peristigmatic gland which is present around the spiracle in case of dipteran larvae and which are hydrophobic in nature. Then what is the function of this epidermal layer? Epidermal layer secrete this topmost cuticle layer. This epidermal layer or epidermis digests and absorbs the old cuticle during the process of molting. Epidermis help in wound repairing in insect and gives surface look to the insect. We have completed about cuticle and its different layers: epicuticle, exocuticle, endocuticle, and we also completed about epidermal layer. Now we are going to discuss about basement membrane. Basement membrane is the lowest or lowermost layer of the insect body wall. It is amorphous, granular, and neutral mucopolysaccharide layer. Neutral mucopolysaccharide layer of thickness is 0.5 microns and it lies just below the epidermis and it forms a continuous sheet similar to that of neural lamella. Then what is endoskeleton? See, this is the epidermal layer. See, this is the cuticular layer. This is the epidermal layer. Below this basement membrane is there. We are seeing here cuticular ingrowth. Here also we are seeing that cuticular ingrowth. So, cuticular ingrowth of the body wall, which provide space for muscle attachment, is known as endoskeleton. Endoskeleton, it is of two types, that is epidermis and apophysis. Hollow invagination of body wall is known as epidermis and solid invagination of the body wall is known as apophysis. What is the function of the body wall? Body wall act as external armor and provide strength to the external organs. It protects the organs against physical abrasion, injurious chemicals, parasites, predators, and pathogens. Internally, it protects all the vital organs such as digestive system, reproductive system, respiratory system, and nervous system. Body wall, it provides space for muscle attachment and gives shape to the body. Body wall, it prevents the water loss from the insect body. Over the insect body, several cuticular sensory organs are there which help in sensing the surrounding environment. Cuticular pigment gives color to the insect body. These are the functions of insect body wall. Now we are going to discuss about cuticular appendages. Cuticular appendages are of two types. They are non-cellular cuticular appendages and cellular cuticular appendages. Non-cellular cuticular appendages have no epidermal association but which are rigidly attached to the body wall. Example for this are minute hairs and thorns. Cuticular appendages, cellular cuticular appendages have epidermal association and which are again two types, unicellular and multicellular. See here, unicellular cuticular appendages are clothing hairs, plumose hairs in honeybees, bristles in flies, scales in moths and butterflies, glandular setae in caterpillars, sensory setae in most of the insects, seta also in most of the insects. Seta, it is a, see here, seta, it is a hair-like outgrowth. Epidermal cells which produce seta is known as trichogen cell. The socket which form house for trichogen cell is known as termogen. Study of arrangement of seta is known as ketotaxy. Ketotaxy. So now we are going to discuss about Multicellular cuticular appendages. They are movable spurs and immovable spines. Now I am going to show you images for all these unicellular and multicellular cuticular appendages. See, clothing hairs and plumose hairs are present in honeybees. See the hairs. Bristles. It is a unicellular cuticular appendages present in flies. Scales. Flattened outgrowth of the body wall present in moths and butterflies. Glandular setae. It is present in caterpillars. Sensory setae, which is present in most of the insect, which is associated with the sensory neurons. Seta, hair-like outgrowth in most of the insects. See here, it is also one of the unicellular cuticular appendage. Now we are going to see multicellular cuticular appendages are spurs. These are the spurs which are movable structure. Spines, these are the spines which are immovable structures. Insect body wall not only associated with the cuticular appendages, it also possesses some glands. Cuticular glands are either unicellular or multicellular. Following are the some of the example. Wax gland present in honeybees and mealybugs. Lac gland is present in lac insect. A molting gland is present in most of the insect which help in the secretion of the molting fluid. Androconia gland or scent cell. Scent cells are present in moths. Poison glands are present in slug caterpillar. See the images. Wax gland of the honeybees here, and mealybug also possess wax gland. See the lac glands in lac insects. Molting gland present in most of the insect help in the secretion of the molting fluid. Androconia gland present in moths. Poison glands present in slug caterpillars. Thank you.
Good morning, my dear students. In this session, we are discussing about molting in insect. Molting, it is also known as ecdysis. See here, this is the insect body wall. We already discussed about insect body wall in previous lecture. Insect body wall consisting of three layers, that is cuticle, epidermis, and basement membrane. Cuticle is again divided into three layers, that is one epicuticle, second one exocuticle, and third one is endocuticle. Knowledge about insect body wall is must in order to understand a molting process in insect. See, then what do you mean by molting? See here, silkworm larva discarding its old cuticle and it's developed new cuticle. Here also, see in this cockroach, the cockroach discarded its old cuticle and it's developed a new cuticle. Therefore, periodical process of shedding the old cuticle accompanied by the formation of the new cuticle is known as molting or ecdysis. The cuticular part discarded during molting is known as exuviae. Okay, and molting occurs many times in insect during its immature stage before it attains the adult stage. Next, see here, this is the silkworm larva. After hatching, first instar will come. This first instar undergoes first time molting and gives second instar. Second instar undergoes second time molting and gives third instar. Third instar undergoes third time molting and gives fourth instar. Fourth instar undergoes fourth time molting and gives fifth instar. See here also, here it is the egg. Egg after hatching it gives first instar. First instar undergoes first time molting and gives second instar. Second instar undergoes second time molting and gives third instar, and so on. See here, this is a one time molting, this is another time molting. So the time interval between two subsequent molting, the time interval between two subsequent molting is known as stadium. And the form assumed by the insect in any stadium, this is the form assumed by the insect. The form assumed by the insect in any stadium is known as instar. Okay. Next, control of molting in insect. In insect, molting is controlled by the endocrine gland, like prothoracic gland. This is the prothoracic gland. This is the prothoracic gland. So, in insect, molting is controlled by the endocrine gland, like prothoracic gland. This gland secretes a molting hormone. See here the steps involved in the process of molting. First, see here, these are the neurosecretory cells. This neurosecretory cell secretes PHTH or brain hormone. PHTH is the prothoracicotropic hormone. This prothoracicotropic hormone activates prothoracic gland. Upon activation, this gland secretes molting hormone, that is 20-hydroxyl-ecdysone. 20-hydroxyl-ecdysone. This molting hormone activates the molting gland which is present in the epidermal layer. Molting gland, upon its activation, it secretes molting gel. Molting gland secretes molting gel. This molting gel during the course of time changed into the molting fluid, and this molting fluid contain activated chitinase and protease that digest old endocuticle and help in the molting of insect. See the steps involved in the process of molting in insect. The insect which undergoes molting, they show behavioral changes. See here, this is the image indicating molting pose in mulberry silkworm. So the following behavioral changes we can observe. Larvae stop feeding and it become inactive. They also show reduced movement, increased grooming, and they seeking out a safe and secluded location. These behavioral changes we can see. During the molting process, we can also observe changes in epidermis. Epidermal cell size. See, this is the epidermis. Here, epidermal cell size, cells activity, protein content, and enzyme content will increases. And here the cells divide mitotically and increases the tension, which result in the losing of cuticle from the epidermal layer. And these changes in epidermal process allows for the growth and regeneration of the new exoskeleton, that is the outer protective layer of the insect body. Next step is apolysis. See here, this cuticular layer is detached from the epidermal layer. It is nothing but apolysis. Due to mitotic division and increased tension in epidermal layer, cuticular layer will detach from the epidermal layer. This is known as apolysis. Due to the apolysis, a space is formed. The space is known as subcuticular space. In this space, molting glands secrete molting gel. which is rich in inactive chitinase and protease. After this, new epicuticle will form, the cuticular layer will be formed, and this cuticular layer protect the epidermal cell and newly forming cuticle from the digestion by the enzyme in the molting gel. Then after procuticle also forms, but here it is not differentiated into exo and endocuticle, it will be differentiated at the last. Next, after formation of protective epicuticle and procuticular layer, the molting gel is activated into molting fluid. Now, the chitinase and protease enzyme, which are present in the molting fluid, begin to digest the old endocuticle. See here, this light blue color portion is there now. This is the endocuticle. It is digested by the molting fluid. As much as 90% of the endocuticle is digested and reabsorbed by the epidermal cell to form procuticle. So this is reabsorbed. That endocuticle is reabsorbed by the epidermal cell to form new cuticle. Okay. The exocuticular layer. See, this is the exocuticular layer. Okay, it contain exocuticle plus epicuticle. The old exocuticular layer, which is resistant to enzyme action since it is highly sclerotized and eventually discarded. This is nothing but exuvial. After this, wax layer will be formed. Pore canals secrete the wax layer. The wax layer serves to reduce water loss and protect the insect from environmental stresses. Next, cement layer will be formed. Cement layer is secreted by the dermal glands, which are also present in the epidermal layer of the insect body wall. సార్ క్లాస్ జరుగుతుంది వస్తాం క్లాస్లో ఉన్నా ఓకే ఓకే next process is molting. Wait for 1 minute. Next process is molting. This involves two steps, that is rupturing of the old cuticle and removal of the old cuticle. Rupturing of the old cuticle and removal of the old cuticle. For rupturing of the old cuticle take place through intake of air or water, which enhances the blood flow to the head and thoracic region. So this increased blood flow will rupture the old cuticle along predetermined line of the weakness known as ecdysial line. It will be ruptured at ecdysial line. Next, removal of old cuticle. After rupturing of the old cuticle, the peristaltic movement of the body, that is contraction and expansion and lubrication action of the molting fluid, will help in the removal of the old cuticle. See here, This peristaltic movement removed the old cuticle. During each molting, the cuticular covering of the body, legs, internal lining of the foregut, hindgut, and trachea are discarded. In the last development of exocuticle and endocuticle, I already told you that procuticle formed before only. Now the procuticle is differentiated into the two layers, that is exocuticle and endocuticle at the last. So the procuticle, which is the primary layer of the insect cuticle, which is developed into both exocuticle and endocuticle during the molting process. Exocuticle, it is formed from the outer region of the procuticle during the molting. It is typically harder and more sclerotized layer than endocuticle and provides structural support and protection to the insects. Endocuticle, this is the inner layer of the insect cuticle, and it's formed from the inner region of the procuticle, and it is generally softer and less sclerotized than the exocuticle and serve mainly as a flexible support layer. Like that, insect discard and develop new cuticle. So the periodical process of removing the old cuticle accompanied by the formation of the new cuticle is the molting in insect. Thank you.
Good morning, my dear friends. In this session, we are going to discuss about structure and modification of insect legs and structure of male and female genitalia. Let's see about insect legs. See, this is the insect thoracic region. Insect thoracic region consisting of three segments, that is prothorax, mesothorax, and metathorax. Each thoracic segment bears a pair of segmented legs. That is, prothoracic legs are known as foreleg, mesothoracic legs are known as midleg, and metathoracic legs are known as hind legs. Therefore, in almost all insects, all the three thoracic segments bears a pair of segmented legs. See, this is the individual leg. Each leg consisting of 5 segments. They are coxa, trochanter, femur, tibia, and tarsus. See, this is the coxa. This is the first segment of the insect leg, and it is freely movable. And this is the trochanter, that is the second segment of the insect leg, usually small and single segmented. And this is the insect leg femur. This is the largest and stoutest part of insect leg. This is the tibial part of insect leg. This is the strongest and longest part of insect leg, and which is provided with these spines and spurs. Tarsus. This is the last segment of the insect leg, and which is end in pretarsus. Then what do you mean by pretarsus? Beyond the tarsus, there are several structures are commonly known as pretarsus. The tarsus terminate into a pair of strongly curved claws, and in between the claws, a median pad-like structure is known as aroleum. At the same time, at the base of the claws, a pair of pad-like structures are there known as pulvilli. These leg pads are useful for insect when it is walking on the smooth surface, while the claws gives needed grip when the insect is walking on the rough surface. When one structure is using by insect, another structure bends upward. See different types of insect legs. In insects, legs are modified into several types based on the habitat and the food habit of the insects. In relation to the habitat, when the insect is aquatic, they develop aquatically suitable legs. When the insect is terrestrial, then they develop terrestrially suitable legs. In relation to the habitat, it's okay. In relation to the food habit, when the insect is predatory, then it's develop the legs in accordance with the predation. When the insect is herbivorous, then it's developed some other kind of leg. Now we are going to see different types of insect legs and their modifications. Ambulatorial legs. These are the walking legs. The fore and mid leg of the grasshopper are the ambulatorial leg, and these legs help in walking for the grasshopper. Saltatorial legs. These are the leaping or jumping leg. The hind leg of grasshopper is the saltatorial leg, which helps the grasshopper for jumping. Cursorial leg. These are the running legs. All the three pairs of legs of cockroach and ants are the running legs. These legs are suited for running in these insects. Scansorial legs. These are the climbing or clinging leg. All the three pairs of legs of head louse are scansorial legs, which help the head louse for climbing or clinging the hairs of host. Fossorial leg. These are the digging or burrowing legs. Four legs of the mole cricket are the fossorial legs. Raptorial leg. These are the predatory or grasping legs. The four legs of the praying mantids are raptorial legs. Natatorial legs. These are the swimming legs. The hind legs of the water bug and water beetles are natatorial legs. These legs help in swimming. These legs help in swimming of the insects. Stick legs. All the three pairs of legs of housefly are the stick's leg. The sticking leg help in attachment of the housefly to the substratum. Clasping leg. Four legs of the male water beetles are the clasping legs. These legs help in clasping the mate during the copulation. Basket legs. The legs of dragonfly and damselfly are the basket legs. These insects capture the prey and keep in their basket and feed on them later. Forageal legs. These are the food collecting legs. The hind legs of honeybees are the forageal legs. In honeybees, the four legs are useful for antennal cleaning. Mid legs are useful for wax picking, and hind legs are useful for food collection. The hind legs of honeybees is provided with the pollen basket, pollen packer, and pollen comb. Now we are going to see legs in immature stage. Immature stage insect consisting of two types of legs. They are thoracic legs or true legs, abdominal legs or prolegs or pseudolegs. The thoracic legs are calling has two legs because these legs come into the adult stage. The abdominal legs are known as pseudolegs because they last in larval or pupal stage only. They do not come in adult stage. The abdominal prolegs are unjointed, short, fleshy structures which are provided with the flat surface at the bottom is known as the planta. A number of hook-like structure on the planta is known as crochets, and these crochets help in clinging and crawling on the surface of the substratum. These crochets are absent in softfly larvae, hence the softfly larvae fall easily when we touch it. Larval classification based on the position of the proleg. See, this is the insect head and thorax. Generally, the thoracic legs are three pairs. These are commonly present in all insect larvae. But see in the abdomen, a pair of proleg is present in the 6th and 10th abdominal segment. Due to the absence of the legs in between the thoracic and 6th pair of abdominal leg, this insect forms a big loop when it is walking. That's why we are calling it as looper. See here, this is the insect head and three pairs of thoracic legs. And this insect containing proleg on 5th, 6th and 10th abdominal segment. Due to the absence of the legs in between thoracic and 5th abdominal leg, this insect forms a small loop-like structure when it is walking. That's why we are calling it as semi-looper. In caterpillars, the prolegs are present on 3rd, 4th, 5th, 6th and 10th abdominal segments. Now we are going to see about female genital structure. Female genital structure is situated on the ventral side of 8th and 9th abdominal segment. The female genital structure or ovipositor contain three pairs of valves or valvulae. See here, this is the first pair of valve, second pair of valve and third pair of valve. Actually, from the base of the 8th segment, valve for 1 will be generated and it gives off a pair of valvulae. That is the first valve. Similarly, from the base of the 9th segment, valve for 2 will be originated and it gives up each of a pair of valvulae 2 and valvulae 3. Valvulae 2 and valvulae 3. See, this is the valvulae 2 and valvulae 3 pairs. See here also in vertical section, this is the first pair of valve. This is the first pair of valve or valvulae, and this is the second pair of valve or valvulae, and this is the third pair of valve or valvulae. Now we are going to see about male genital structure. This is the male genital structure of the click beetle, which is belong to the order Coleoptera and family Elateridae. The whole organ, the whole copulatory organ of higher insect is known as adigus. This adigus helps in insemination. In addition to the insemination, it also helps in clasping and providing sensory stimulation to the female insect. Typically, the adigus contain a tubular structure is known as penis. Penis internally contain a tube-like structure is known as endophallus, and this endophallus is aborted during the insemination. The ejaculatory duct opens at the tip of either penis or endophallus. The lateral lobe-like structure, to the penis are known as the parameres, and these parameres help in clasping and providing sensory stimulation to the female insects. See here the ninth abdominal segment of the bristle thigh and their reproductive organs. Thank you. Good morning, my dear friends. In this session, we are going to discuss about insect antenna. Insect antenna are also known as feelers because they feel the surrounding environment. Insect antennae are paired, highly mobile and segmented structures. Antennae are located between or behind the compound eyes. See here a pair of antennae which are locating in between the compound eyes. The antenna is set in a socket of the cranium is known as antennal socket. The base of the antenna is connected to the edge of the socket by means of an articulatory membrane, and this membrane permits the free movement of antenna. Antenna consisting of three parts, they are scape, pedicel and flagellum. The basal segment of the antenna is known as scape. It is conspicuously larger than succeeding segments. The second segment of the antenna is known as pedicel, which immediately follows the scape. The pedicel bears a mass of sense cells known as Johnston organs, and these Johnston organs are used as cardotonal organ in some of the insects like mosquitoes. Cardotonal organs are the structures which detect a wide range of mechanical stimuli. Both scape and pedicels are provided with intrinsic muscle. The scape and pedicel provided with intrinsic muscle. Intrinsic muscles are the muscles of the body that occurs deep within the structures. The remaining antennal segments are combinedly known as flagellum or clavola, which lacks individual muscles. Surface of the flagellum is provided with many sensory receptors that are innervated by the deuterocerebrum of the brain. Flagellum may vary in size and form in different insects. Functions of antenna. Antenna is useful to detect chemicals, including food and pheromones. Pheromones are the chemicals secreted into air by one sex to attract opposite sex, and it is generally secreted by female insects. Antenna perceives smell, humidity changes, variation in temperature, vibration, wind velocity, and direction. Antenna is useful to pursue the forward environment and detect danger. It is useful for hearing in mosquitoes and communication in ants. Rarely, it is also useful to clasp the mate in fleas and grasp the prey in some other insects. Types of antenna. There are several types of antennas. Now we are going to discuss one by one. Cetaceous or bristle-like antennae. In this type of antennae, the size of the segment decreases from base to the apex. See here, basal segments are somewhat larger and its size is decreasing from base to the apex. Cetaceous antennae found in cockroach, leafhopper, dragonfly and damselfly. Filiform antennae are thread-like antennae. In filiform antennae, the antennal segments are usually cylindrical and thickness of segment thickness of segments remains same throughout the length of the antenna. And this filiform antennae found in grasshoppers. Moniliform antennae are beaded antennae. In this type of antennae, the antennal segments are either globular or spherical with prominent constriction in between the segments. Moniliform antennae found in termites. See, this is the moniliform antenna. Serrate antenna or saw-like antenna. In this type of antennae, the antennal segment have short triangular projection on one side. Serrate type of antenna found in jewel beetle and longicorn beetle. Unipectinate antenna or one-side comb-like antenna. In this type of antenna, the antennal segment bears long, slender lateral processes on one side. Unipectinate antenna found in sawflies. Bipectinate antennae or two-side comb-like antenna. In this type of antenna, the antennal segment bears long, slender lateral processes on both sides. See the lateral processes on both sides. Bipectinate antennae found in silkworm moth. Clavate or clubbed antenna. In this type of antenna, the antennal segment enlarges gradually towards the tip. See here, antennal segments size is increasing gradually towards the tip. And clavate antenna is found in blister beetle. Capitate or knobed antenna. In this type of antenna, the terminal segment become enlarged suddenly. See here, the terminal segments enlarge suddenly. Capitate antenna is found in butterflies. Lamellate or plate-like antenna. In this type of antenna, the antennal tip is expanded laterally on one side and form plate-like structure. See here, plate-like structure. Lamellate antenna or plate-like antenna found in lamellicorn beetle. Aristate antenna. In this type of antennae, the terminal segment is enlarged and bears a conspicuous dorsal bristle known as arista, and the aristate antenna found in houseflies. Stylate antenna. In this type of antenna, the terminal segment bear a style-like process. That's why we calling it as stylate antenna. Stylate antenna found in horsefly and robberfly. Plumose or feathery antenna. Antenna is highly feathery and antennal segment bears long whorls of hairs. Plumose antenna found in male mosquito. Pilose or hairy antenna. Antenna is less feathery with few hairs at the junction of flagellomeres. Pilose antenna found in female mosquitoes. Geniculate or elbowed antenna. In this type of antenna, the scape is long and remaining all the segments are small and which are arranged at an angle to the first segment, that is scape, and gives the appearance of an elbow joint. Geniculate antenna found in ants, weevils, and honeybees. Flabellate are fan-like or tongue-like antenna. In this type of antennae, the segment of flagellum are modified into long, thick, fan or tongue-like projections. Flabellate antennae found in Strepsiptera stylopids. All insects except Procera possess a pair of antennae, that is, in Procera antennae are absent. Antennae are also absent in class Arachnida. Procerons are the blind insects because they do not possess any eyes and antennae. Two pairs of antennae are present in crustaceans. Antennae are well developed in adults and poorly developed in immature stages. Johnston organs are absent in Typlura and Columbola. Thank you.
Good morning, my dear friends. In this session, we are going to discuss about a respiratory system in insects. In any organism, respiration is the most important process because which helps in cellular oxidation of the glucose molecule and yields ATP molecule. And this ATP molecule has a very essential role in maintaining metabolism, body temperature, different body processes such as digestion, circulation, excretion, etc. Like aerobic animals, Insects also respire by taking oxygen from their surrounding environment and eliminating carbon dioxide respired by their cells. In insect, air is directly supplied to the tissues, and hemolymph is not involved in the process of respiration, as in case of human being. Important structures involved in insect respiration are trachea and spiracles. See here the tubular structure which involved in the gaseous exchange are known as trachea. See, this is the trachea and it gives very fine branches known as tracheoles. Spiracles. See the small opening, that is tracheal outside opening is known as spiracles. Spiracles are situated laterally on the body wall. No insect has more than 10 pairs of spiracles, that is 2 pairs in the thoracic region and 8 pair in the abdominal region. See here the spiracles of grubba rhinoceros beetles. These are the abdominal spiracles and this is the thoracic spiracles. In insect, exchange of gases take place through the tubular structure is known as trachea. These trachea are distributed throughout the body and form tracheal system. The trachea opens outside on the body wall through a small opening is known as spiracles. See the spiracles. The spiracle is surrounded by a small ring-like structure is known as peritrium. The spiracles lead into a cavity is known as atrium. This atrium is provided with the atrial wall which help in the closing and opening mechanism of spiracles. Atrial wall also provided with the trichomes. This help in filtering out the dust particles. Trachea are fine elastic tubular structures and ectodermal in origin and are made up of 3 layers, that is outer basement membrane, middle epidermis and inner cuticular layer. The cuticular lining inside form intima. The spiral thickening of the intima inside the tube is known as ctenidia, and this ctenidia avoids the collapse of trachea when there is no air. Terminal cell of the trachea is known as tracheoblast or tracheolar cells. This tracheoblast gives very fine branches are known as tracheoles. Of size is less than 1 micron in diameter. These tracheoles penetrate the cells and intimately associated with the mitochondria. Longitudinal tracheal trunks. The trachea coming from the spiracle throughout the body joins with the trachea of the neighboring spiracle and forms longitudinal tracheal trunks. In insect, there are 4 longitudinal tracheal trunks, that is 1 dorsal trunk which help in supplying oxygen to the proximal part of the body as well as to the heart. Two lateral trunks which supply oxygen to the alimentary canal, gonads, legs, and wings. Ventral tracheal trunk which supply oxygen to the central nervous system. Among all these trunk, lateral longitudinal trunks are longest one, which are extended in thoracic and head region also. In absence of ctenidia in trachea, thin-walled collapsible sac-like dilation is present, is known as air sac. Air sac acts as oxygen reservoir, provides buoyancy to the flying and aquatic insects, provides space for growing organ, act as sound resonator and heat insulators. Mechanism of respiration. Oxygen enters the spiracles, passes through the trachea and tracheoles, and finally reaches to the target cells by a combination of ventilation and diffusion along a concentration gradient from high in the external air to the low in the tissues. The net movement of the oxygen molecule into the tracheal system is known as inspiration, whereas the net movement of the carbon dioxide and water vapor molecule outside of the tracheal system is known as expiration. Classification of tracheal system. Based on the number and location of functional spiracles, respiratory system is classified as follows. Holophenastic, all the 10 pairs of spiracles are functional. Hemiphenastic, out of 10 pairs, 1 or 2 pairs are non-functional. Hypophenastic, 1 to 2 pairs of spiracles are completely absent here. Hyperphenastic, presence of more than 10 pairs of spiracles are functional. Holophenastic, see this blue color dot indicate functional spiracles. In holophenastic respiratory system, all the 10 pairs of the spiracles are functional. This is the case in case of grasshopper, cockroach, and dragonfly. Hemiphenastic, in hemiphenastic, out of 10 pairs, 1 or 2 pairs of spiracles are non-functional. See, this red colored dot indicate non-functional spiracles. Hemiphenastic type of spiracles are following types. Periphenastic, that is peripheral are functional. This is the middle one, and this peripheral are functional. This is the case in case of larvae of Hymenoptera and Coleoptera. Amphiphenastic, anterior is functional, posterior is functional, and middle one are non-functional. Amphiphenastic, anterior posterior functional in the case of larvae of Cyclorapha. Prophenastic, first one is functional, remaining all are non-functional. This is the case in case of tumbler and puparium. Metaphenastic, that is the last pair of spiracle is functional and all other spiracles are non-functional in case of wriggler. Aphenastic, here all the spiracles are non-functional. This is the case in case of larvae of maximum flies and nymphs of Odonata. Hypophenastic, 1 or 2 pair of spiracles are completely absent. This is the case in case of Mellophaga and Siphunculata. Hyperphenastic, see here green color spiracle. It is extra in prothoracic region of insect body. That is presence of more than 10 pair of functional spiracles in diplurans is the hyperphenastic condition. Hyper means somewhat extra. So here more than 10 pairs, that is 11 pairs of the spiracles are functional. Other type of respiration, cutaneous respiration take place in endoparasitic insect, Procura and Columbola. Tracheal gill respiration is take place in mayfly needs. They have lateral gills. Damselfly needs they have caudal gills, and dragonfly needs they have rectal gills. Air store is present in case of water bug. Respiratory siphon is present in regular. Caudal breathing tube is present in water scorpion, and plastron type of respiration is present in aquatic beetles. Thank you.
Good morning, my dear friends. In this session, we are going to discuss about excretory system in insects. In insects, excreta is produced by means of two related processes. They are excretion and osmoregulation. Then what do you mean by excretion? The removal of waste product of metabolism, especially nitrogenous compound, from the body of the insect is known as excretion. Excretion helps in avoiding the toxic effect of the nitrogenous compound on the insect body. Then what is osmoregulation? A homeostatic mechanism that regulates the optimum concentration of water and salts in the tissues and body fluid is known as osmoregulation. Osmoregulation helps in the maintaining the internal environment of the insect body. Then what is excretory system? The system responsible for excretion and osmoregulation is known as excretory system. The waste product excreted may be in different form, that is, may be in solid form, that is urea or uric acid, semi-solid form, that is allantoin, liquid form, that is honeydew, gaseous form, that is carbon dioxide, amide form, that is ammonia. The organisms which excrete urea or uric acid are known as uricotelic organisms. The organisms which excrete ammonia are known as ammonotelic organism. Example is most of the aquatic animals. See insects. Insects are uricotelic. They produce relatively dry waste matter, mainly uric acid. It is an important adaptation to the terrestrial life to conserve the loss of water. Organs of excretions include Malpighian tubules, body valve, tracheal system, alimentary canal, labial glands, fat bodies, nephrocytes, enocytes, and chloride cells. Malpighian tubules. Long, thin, tubular outgrowth of the alimentary canal at the junction of midgut and hindgut is known as Malpighian tubules. Malpighian tubules are generally free, waving around in the hemolymph with distal secretory region and proximal absorptive region. Malpighian tubules are the major organ of excretion. They help in removal of nitrogenous waste product from the hemolymph and help in osmoregulation of the insect body. Malpighian tubules are discovered by Marcello Malpighi and named by Haeckel. Malpighian tubules are absent in springtail and aphids, 2 in numbers in scale insects, 4 in bugs, 5 in mosquitoes, 6 in moths and butterflies, 60 in cockroach,
and more than 200 in locusts. Physiology of Malpighian tubule. Due to the active movement of the potassium ion into the tubule lumen generate an osmotic pressure gradient in the distal end of the Malpighian tubules, result in passive movement of the water, amino acid, sugars, salt, and active movement of nitrogenous waste material occurs in the distal secretory region of the Malpighian tubule and forms primary urine. Due to the continuous secretory activity of the distal region, this primary urine moves forward into the proximal and absorptive and finally into the gut region. During the process of transportation in the proximal region, due to the acidic pH, the uric acid is precipitated and the important materials such as sugar, water, potassium, sodium, and chlorine are reabsorbed and returned back to the hemolymph. Finally, the secondary urine, uric acid, alkaloids, ammonium salt, and other toxins are excreted out through the anus. Cryptonephridial condition. Attachment of the distal end of the Malpighian tubule with the rectal wall by means of perimetric membrane. Attachment of the distal end of the Malpighian tubule with the rectal wall by means of perinephric membrane is known as cryptonephridial condition. This is concerned either with the efficient dehydration of the fecus before their elimination or ionic regulation. Cryptonephridial conditions seen in adult Coleoptera, larval Lepidoptera, and larval Symphyta, that is softfly. Functions of the Malpighian tubules. Malpighian tubules are the major organ of excretion. They perform excretory function. Beside excretion, they also perform following accessory functions. They are spittle secretion in spittle bug, light production in Boletophila, production of stalked egg in case of aphid lion, and Malpighian tubule also store calcium, which is necessary for hardening of the puparium. Other excretory organs are the body wall. Body wall is the outer covering of the insect body. Through the process of molting, body wall removes the exuvia. It contains the nitrogenous waste product. In some of the endoparasitic insect, through the cutaneous respiration, elimination of the carbon dioxide occurs. Tracheal system. Tracheal system help in the elimination of carbon dioxide through the spiracles. Alimentary canal. Alimentary canal help in digestion and absorption of important material and also help in the excretion of undigested waste materials. Labial glands. Sac-like ampulla help in the excretion of the ammonia in Thysanura, Dipleura, and Columbola. Fat bodies, nephrocytes, enocytes. These all are hemocytes present in the hemolymph of the insect and help in the storage excretion of the nitrogenous waste material. In freshwater insects such as needs of dragonflies and damselflies, special cells are known as the chloride cells which are responsible for the excretion and osmoregulation. Storage excretion. Because uric acid is so insoluble, it can be easily stored in different sites of the insects without its interaction with the other physiological processes. The excretory waste material, that is uric acid, are retained within the insect body in different sites, such as cells of the fat body in American cockroach, in body wall of the red cotton bug, and this will gives white color to the red cotton bug. Male accessory gland, in male accessory gland in several insects, in wing scales of period butterfly, and it gives white color to the wings of the period butterflies. Uric acid also stored in waste product of pupal metabolism. Like this, the uric acid is stored and later excreted. That's why we calling it as storage excretion. Thank you.
Good morning, my dear students. In this session, we are going to discuss about bee dances. Before going into the discussion, let's see what do you mean by bee flora or bee pasturage or bee forage. See here number of plant species which are providing pollen, propolis and nectar to the bees. These all are commonly known as bee flora or bee pasturage or bee forage. Then what do you mean by bee foraging? The act of searching food by the honeybees is known as bee foraging. See here, this is the artificial honey hive. This honey hive accommodate around 8 to 10 frames, and honeybee colony will stay in this honey hive. Honeybee colony consisting of a single queen, hundreds of drones, and thousands of workers. Then, what is the role of queen bees in the colony? See, this is the queen bee. This is the only fertile female in the colony which is responsible for laying eggs and ensure the colony's survival and growth. These are the drone bees and these are the male bees whose sole purpose is to mate with the queen bees and making them fertilize. In the honeybee colony, thousands of workers also there. These are the sterile female. They perform various indoor and outdoor duties like cleaning up the hive, nursing the larvae and queen, guarding the hive, collection and storage of the food, maintaining the temperature of the colony for community survival. For all these things, effective communication among the bees is essential. Therefore, honeybees communicate with each other by means of two ways. That is, one, the physical communication by means of doing dances, chemical communication by means of producing pheromones or chemicals. In this session, we are discussing about physical communication by means of bee dances. Bee perform two types of dances, that is one round dance and second one is wagtail dance. See here, this is the honey hive and this is the bee forage. And the distance between the honey hive and bee forage is less than 50 meter. In this case, when a food source is located very close to the hive, when a food source is located very close to the beehive, that is less than 50 meter away, the forager bee perform a round dance. And this round dance indicates only the distance of the food, but it do not indicate direction of the food. The forager bee perform a round dance by running around in a narrow circle and suddenly reversing its direction to its original position. And she may repeat the dance several times at the same location or move to the another location to perform the same dance. After the completion of the dance, she often distributes the food to the follower bees so that the follower bees visit the same food source, they collect the food and return back to the hive, and there they store their food. See here, in case of round dance, the forager bee is going for foraging. After finding the bee forage, it collects pollen, propolis, and nectar and return to the hive. And on the surface of the frame, it performs a round dance. After completion of the round dance, upon receiving information, the follower bees moves out of the hive and goes to the same forage. They collect the pollen, propolis, and nectar and return back to the hive, and there they store their food, that is collected food. Like that, the round has indicate only the distance of the food. Now you see here waggled hands. See, this is the honey hive and this is the bee forage. When bee forage, see here the distance between the honey hive and bee forage is greater than 50 meter. When a food source is distantly located to the beehive, that is more than 50 meter away, a forager perform wagtail dance. In wagtail dance, the bees or the forager bees run straight ahead for shorter distance by making waggle and return to its original position by making a semicircle. Again, the bee moves in straight line for shorter distance by making waggles and run or make another semicircle in opposite direction. Like that, the forager bee will complete a full figure of 8 circuit. While running in the straight line, the vibration in the abdomen gives the tail waggling motion, and this wagtail dance gives or this wagtail dance communicate both distance and direction to the follower bees. See here, in case of waggle dance, the forager bee is going for foraging. Once it finds bee forage, it collect it and return back to the hive, and on the surface of the hive it perform a wagtail dance. Upon receiving information, the follower bees move to the same forage. They also collect the forage and return back to the hive where they store their food. I told you that a wagtail dance indicates both distance and direction. See here how this dance will indicate distance. In the straight line, when the bee makes less number of waggle, means it indicates the food source is nearer. In the straight run, when bees making several number of waggle, mean it indicate the food is somewhat far. Another indication is When bees are making small and several number of circuits, then it indicates the food is somewhat nearer. When the bees are making very few number of circuits and large circuit, very few number of circuits, and their size is large, it indicates the food is somewhat far. I also told you that this wagtail dance also indicate direction. Yes, this dance indicate direction. See here, this is the honey hive, this is the food source, and this is the sun. When the food is in the direction of the sun, the bee sits at the bottom of the frame and moves vertically upward. Moves vertically upward. See here, this is the honey hive, and this is the food, and this is the sun. Here the food is in the opposite direction to the sun. Therefore, in this case, the bees sits at the top of the hive and moves vertically downward. It indicates the other follower bees, the food source is away from the sun, so that they move in the opposite direction to the sun and they find their food. When the food is in the 60 degrees angle to the sun, the bee performs waggle run 60 degrees angle to the straight line. Like that, the direction of the food is indicated in the waggle run. For the interpretation of the meaning of this dance, Karl von Frisch awarded with the Nobel Prize. Thank you.
Good morning, my dear students. In this lecture, we are going to discuss regarding study of different species of mulberry and non-mulberry silkworms. There are two types of silkworms. That is, one, mulberry leaf-eating worms are known as mulberry silkworm. Non-mulberry leaf-eating worms are known as non-mulberry silkworm. They are Tazark silkworm, Muga silkworm, and Eri silkworm. First, we are going to discuss about mulberry silkworm. Mulberry silkworm scientific name is Bombyx mori, and it's belong to the family Bombycidae. Mulberry silkworm is domesticated for more than 4,000 years. It is being cultivated on mulberry leaves. The moths are milky white in color, medium sized, with smoky wavy line on white colored fore and hind wings. That is, smoky wavy lines on white colored fore and hind wings. The apex of the forewing is slightly elongated and curved. Frenulum, that is the wing coupling apparatus, is almost atrophied or absent. Maxillary palpi and tymphanal organs are absent, but bipectinate antennae are present. These are the mulberry leaves. This is the mulberry silkworm larvae, and this is the adult. See the wavy lines on fore and hind white wings of silkworm moth, and it possesses bipectinate antennae. Larva in mulberry silkworm is glabrous, that is without hair, and elongate with a mid-dorsal caudal horn and eighth abdominal segment. Cocoons vary in shape and color according to the races. The cocoon composes of a single continuous reliable thread which is ranging from 500 to 1500 meter in length, and the pupa is uptake type. The mulberry silkworm larva is monophagous and feeds on mulberry, that is Morus indica, Morus alba, Morus rubra, Morus nigra, etc. Now we are going to see about non-mulberry silkworm. First one is airy silkworm. Airy silkworm's scientific name is Samia cynthia resini, or synonym is Phyllosamia resini. It is belong to the family Saturnidae. See here, moth are brownish block in color, large sized with about 8 to 10 centimeter wing expansion. Wings are speckled with triangular eye spot almost near its center with characteristic brown lines. This moth also possesses bipectinate antennae. Larvae are domesticated, polymorphic, robust, and possess coli. See here, spine-like structure are known as coli. Larval period is 25 to 20 to 25 days. It forms creamy white or dark brown, elongated and soft cocoon with an opening at one end. See the opening here, hence called open end cocoon. The silk is non-reliable since it is not in a one single strand, that is not continuous. Pupa is uptake type. It can pupate anywhere, so Chandrika is not required for this silkworm moth. See, this is the airy silkworm moth adult, airy silkworm moth larvae. Airy silkworm is polyphagous and feed on castor, tapioca, champa, kesseru, bar kesseru. Next non-mulberry silkworm is Tazar silkworm. Its scientific name is Anderia mylitta. Moths are large in size with 20 centimeter wing expansion and polymorphic, comes in different colors. The wings of male vary from brown to dark gray in color, while in female the wings vary from yellowish gray to brownish shades. Wings are speckled with transparent circular eye spot near the center. They also possess bipectinate antennae. The larva is green, robust, possesses coli, and is semi-domesticated. The larval period is 30 to 40 days. It forms dense, firm cocoons. The cocoon color varies from grayish to yellowish shades. The filament is reliable and measure around 500 to 1300 meter. Pupa is uptake type. This is the Tazar silkworm moth adult, larvae, life cycle, and the Tazar silkworms are polyphagous and mainly feed on Terminalia tomentosa, Terminalia arjuna, Syzygium cumini, Soria robusta, Gigipus jujuba, Lagestomia parviflora, Tectoma grandis, and Acacia auriculiformis. Another non-mulberry silkworm is Muga silkworm. Its scientific name is Anderia azamensis. Moths are large with 25 centimeter wing expansion. The wings are brown with a transparent eye spot in the center. They also possess bipectinate antennae. Larvae are green in color and semi-domesticated and read mostly in Assam. Cocoon color varies from golden to glossy white with rudimentary peduncle, hence called golden cocoon. The filament is reliable. Pupa is object type. The larva is polyphagous and feed on some and solo plants. See, this is the Muga silkworm adult. Life cycle of Muga silkworm. Some host plant and solo host plant. Thank you.
Good morning, my dear students. In this lecture, we are going to discuss regarding grainage, chalky rearing, and late age rearing of silkworm. Quality seed is the backbone of sericulture industry. Such quality seeds are produced at breeding centers, grainages, and distributed to the farmers or the rearers. In India, usually three-tier system of seed production is adopted, that is at P3 center, P2 center, and P1 center. At P3 centers, basic or nucleus seeds are produced. At P2 centers, foundation stock seeds are produced. P2 center also serve as germplasm bank. P1 centers receives the cocoon from the P2 centers, and the parents of commercial layings are mass multiplied here and distributed to the rearers or the farmers. Silkworm eggs are known as silk seeds. These are of two types, that is one reproductive seed and another one is the industrial or commercial seeds. The reproductive seeds are produced in very limited quantity in special breeding centers by technically qualified persons. The purpose of the production of the reproductive seed is for maintaining the racial purity and to produce parents for the commercial seeds. The commercial seeds, on other hand, produced in large quantity in special organization called grainages. The purpose of production of commercial seed is to supply to the rearers or the farmers. Grainage. See, this is the grainage. Grainage is a center where the disease-free silkworm seeds are produced in large quantity under scientific line. These centers purchase cocoon from the certified seed cocoon producers, allow this cocoon for moth emergence, make moth sexing and obtain large quantity of the seeds. And these seeds are mass multiplied and distributed to the rearers or the farmers. The grainages are mostly owned by the government and may also be owned by licensed private owners. See here, silkworm cocoon. Grainages collect large quantity of silkworm cocoon, allows for moth emergence and make mating of the moth, and allow the moth for laying large quantity of the eggs, and these eggs are mass multiplied and distributed to the farmers. See, this is the plan of grainage building. A grainage building must and should contain following facilities, such as a room for accommodating cocoons or pupa of different strains, a room for coupling and oviposition of the moth, incubation room, laboratory, egg processing room, cold storage room, dormitory, and office room. The location of the grainage must be in cooler areas of the locality. The grainage must be situated in the center of this sericulturally active area, so that most of the people approaches this grainage and take the egg for sericulture. Activities taking place at grainage. The grainage center purchase cocoon from the certified seed cocoon producers. These cocoons are placed in well ventilated rooms which are provided with optimum temperature of 23 to 25 degrees Celsius and humidity of 70 to 80%. And here moth emergence is allowed. These grainage rooms are may be kept at dark, and light may be supplied suddenly on the expected day of emergence to bring uniform emergence of the silkworm moth. Emerging moths are sexed and used for breeding purpose to produce seed eggs. 3 hours of mating secures maximum fertilized eggs. The sexed females are then made to lay egg on paper sheets or cardboards which are coated with a gummy substance. These egg sheets are disinfected with 2% formalin and then washed with the water to remove the traces of formalin and then dried up in the shades. The dried eggs weighed to the required standard and packed in small flat boxes with muslin cover and dispatched to the buyers. Now the silk seeds are in the hands of farmers or the rearers. Farmers incubate them and obtain silkworm larvae. These larvae are provided with good quality food, and grown-up larvae are mounted on the mountages. On the mountages, these larvae form cocoons. Cocoons are harvested and marketed. These marketed cocoons are processed and silk is extracted. See, all this procedure are processed in detail. Egg incubation and hatching. Silkworm rearing starts with silkworm egg incubation. Eggs are incubated under optimum temperature of 25 degrees Celsius and at humidity of 80 to 85%. Under these conditions, eggs are expected to hatch within 10 to 12 days. See here, eggs are
taking into the hatching tray for incubation purpose. A hatching tray accommodates around 30,000 eggs. Here, hatching tray covered with tissue paper. Over it, another plate is placed for proper coverage and incubation. See here several hatching trays kept for incubation in a single large tray. Here block boxing process is going on to ensure uniform egg hatching in silkworm. The eggs at pinhead stage are kept under total darkness either by wrapping them in the block paper or keeping them in a block box or keeping them in a block room for 2 days before hatching. This process is called block boxing. The next day they are exposed to the diffused light so that the larvae hatch uniformly in response to the photoic stimuli. In silkworm, eggs generally hatch in 10 days, so block boxing is done on 8th day or 2 days prior to the egg hatch, result in 90% hatching can be obtained. See here, after block boxing, around 90% hatch larvae on the sheet tissue or on the tissue paper. After larval hatching, they must be transferred to the new tray or rearing beds. There is two acceptable method for larval transfer to the new trays or rearing bed. See first method, this is the first method and this is the second method. In first method, the hatching trays are covered with porous nylon net. Over this net, chopped leaves provided as the feed. To this feed, the hatched larvae are attracted and they crawl over the nylon net. So, the larvae, along with feed, this net is taken to the new tray like that transferred. See, take out nylon net with crawl larvae and transfer to the new tray and give first feed to them. In second method, brushing is done. The process of transferring the silkworm to rearing tray is known as brushing. The hatched worms are brushed with a feather from the egg guard directly onto the rearing bed. Here, care must be taken not to injure the worms and give first feed. See here, first feed is given. Silkworm undergoes 4 molting and gives 5 instars. Molting or ecdysis is nothing but periodical process of shedding the old cuticle accompanied by the formation of the new cuticle is known as molting or ecdysis. The time interval between the two subsequent molting is known as stadium, and the form assumed by the insect in any stadium is known as instar. Now we are going to discuss what is chalky rearing and what is late age rearing. Rearing of first and second instar larvae is known as chalky rearing. Rearing of third, fourth and fifth instar larvae is known as late age rearing. During rearing, temperature and humidity play most important role. Silkworms do well in the temperature range of 23 to 28 degrees Celsius, with higher temperature for the younger worms and lower temperature for the older worms. During rearing, we have to provide around 70 to 85% relative humidity, higher for the younger worms and lower for the older worms. Temperature and humidity can be moderated by use of wet gunny bag or wet newspaper or pouring the water on cemented floors. See in this table optimum temperature and humidity for rearing of different instar of silkworm are given below. For first instar, we have to provide 26 to 28 degrees Celsius temperature. For second instar, same, that is 26 to 28 degrees Celsius. For third instar, 25 to 26 degrees Celsius. For fourth instar, 24 to 25 degrees Celsius. For fifth instar, 23 to 24 degrees Celsius. See here, we are providing somewhat higher temperature to the first instar and somewhat lower temperature to the fifth instar. Similarly, we are providing somewhat higher humidity, that is 85 to 90% to the first instar and lower humidity to the fifth instar, that is 70%. Silkworm density or spacing of silkworms. Different instars of silkworms require different bed space. So, spreads the worm evenly and ensure that they do not overlap each other. Overlapping or overcrowding leads to underfeeding, creation of microclimate for disease spread, and could also lead to the suffocation. See here, rearing density for one case of silkworm given here. For first instar, provide 1 meter square area. For second instar, provide 2 meter square area. For third instar, provide 4 meter square area. For fourth instar, provide 8 meter square area. For fifth instar, provide 17 meter square area. Now we are going to discuss stages of growth and feeding method. In first stage or in first instar, we have to feed the worm with young tender leaves, that is second and third leaves which are collected from tip of the shoot, and like this we have to feed for the worm for 4 days. After this period, the worm go into the molt. What are the signs that silkworm exhibit during molting? The molting signs are swollen heads, raised head, worms are immobile. During molting, the bed should be kept dry and uncovered. Signs of getting out of molt: the worms are active and move around. The mouth part is broader. The body is dull with loose skin. Stud of skin are easily seen on the bed. Once the worm come out of the molt, spread them out evenly on the bed and provide optimum space to match with their increasing body size. In second instar, feed the worm with third and fourth young glossy leaves, and feeding continue for 4 days. After this period, the worm go into the molt again. In third instar, the larvae feed with good leaves harvested from the green part of the plant and continue feeding for 3 days.
After this period, the worm go into the molt again to the fourth instar. In the fourth instar, feed the worm with whole suit and continue feeding for the 6 days. After this period, the worm go into the molt again. In fifth instar, feed the worms with whole suit and feeding continued for 7 to 8 days. After this fifth instar, the larvae are ready for spinning and they are transferred to the mountages for spinning up the cocoons. During feeding, always maintain a single layer of the suit. Avoid overmature, yellow and diseased leaves. There are 3 methods for rearing of late-age silkworms. They are self-rearing, floor rearing, and shoot rearing. Now we are going to discuss all this method one by one. See, this is the self-rearing. These are the selves. Over these selves, bamboo trays are kept. Bamboo trays are more common in Karnataka and West Bengal, of diameter 1.2 to 1.4 meters. See self-rearing by using bamboo and wooden trays. In self-rearing, silkworms are reared in bamboo trays. See here bamboo trays, which are arranged one over the other in trays on a rearing stand. This is the rearing stand. Each rearing stand can accommodate 10 rearing trays. In this system, leaves are picked individually from the plant or branches of mulberry cut into the convenient size and are fed to the silkworms. Usually 4 to 5 feedings are given in a day and nets are used for cleaning the beds. This is the disadvantage and this is the advantage of self-rearing. Since cleaning is practiced more frequently in self-rearing and also tray removed from the stand for every feeding required more number of labors, so it is somewhat costly. However, this method has the advantage of accommodating more silkworm in limited space than the other two methods, and this method is preferred by the Indian farmers who are invariably confronted with the problem of rearing space. Next, we are going to discuss regarding floor rearing. These are the fixed rearing seats of width is 1 to 1.5 meter and the space between two rearing seats is zero point 0.6 to zero point 0.8 meter and the length of the seats is 5 to 7 meter, or it is depend on the size of rearing room. In floor rearing method, the worms are reared on fixed rearing seats. The rearing seats are arranged in 2 or 3 trays for accommodating large number of silkworm as it's possible. The rearing seats may measure 1 to 1.5 meter wide, 5 to 7 meter long according to the size of the rearing room. The space between the two tiers is zero point 0.6 to zero point 0.8 meter and sufficient space is left all around the rearing seat for attending to feeding and to carry out other rearing operations. As in the self-rearing, the silkworms are fed with the leaves or branches cut into the small size. The number of feeding given in this method is 3 to 4 in a day. Bed cleaning is also followed in the same way as for the self-rearing by using nets, but it is done less frequently, twice during the fourth instar and thrice during the fifth instar. The main advantage in this system as compared to the self-rearing is that feeding and cleaning processes do not involve the handling of innumerable trays. As such, there is considerable saving on labors. This is the last method of rearing late-age silkworm, that is shoot rearing. This method is similar to that of floor rearing. Here also silkworm larvae reared on fixed rearing seats, but which are provided with whole shoot. See here, we are giving whole shoot as the feed to the silkworm larvae. Since the leaf supply due to the shoot feeding is distributed in 3 dimensions, there is better aeration of the rearing beds, and therefore it is possible to have 50% more warm per unit area of rearing seat as compared to the self or floor rearing. Cleaning in this method is also reduced to the minimum, which is normally carried out once each in the fourth and fifth instars. See the process for bed cleaning in shoot rearing method. Ropes of convenient length are spread parallel to each other along the length of the rearing bed. Over these ropes, fresh branches of mulberry are supplied for feeding. After 2 or 3 feeds, when all the worms have crawled onto the new branches, the bed held by the rope is rolled into the loose bundle, the old branches and dirt is removed. This is the most economical method of rearing, as labor requirement for the process of feeding and bed cleaning are kept at minimal level. This table showing leaf requirement for rearing of 1 kg silkworm. To the first instar larva, we have to provide second and third young leaves which are collected from the tip of the plant. In first instar, on first day we have to provide 150 gram leaf, on second day 200 gram leaf, on third day 200 gram leaf, on fourth day 150 gram leaves, and on fifth day the worms go into the sleep or mount. Like that, in second instar we have to provide this much quantity of leaf. In third instar we have to provide this much quantity of the leaves. In fourth instar we have to provide this much quantity of the leaves, and in fifth instar also we have to provide this much quantity of the leaves. The most important step in silkworm rearing is the bed cleaning. Bed cleaning is done to remove fecal matter, dead worms and leaf remnants which would promote fungal growth. It is done after every molt for the young silkworm, but every other day for the mature worms. Bed cleaning is best done before feeding the worms. See here the procedure given for bed cleaning. To clean the bed, spread a net over the worm
and spread fresh leaves on the top of the net. Worms will crawl up to feed on the fresh leaves which are present on the surface of the net. Then transfer the worm using the net to another clean bed or the tray. Remove any remaining worms and then discard the waste into a compost pile. Thank you.
Good morning, my dear students. In this lecture, we are going to discuss regarding mounting and harvesting of cocoons. Mounting for cocoon spinning, transferring of mature fifth instar silkworm larvae from rearing beds to the mountages is known as mounting. For cocoon spinning, transferring of mature 15-star silkworm larvae from rearing beds to the mountages is known as mounting. In silkworm larvae, spinning starts 8 days after worms get into the 5th instar stage. When the silkworm larvae exhibits following signs of maturity, then they are ready for mounting. Let's see the signs of maturity in silkworm larvae. Larvae cease to feed and crawl restlessly in search of corner to attach themselves for spinning. They move to the corner of the rearing beds for spinning. If picking up mature silkworm is delayed, then the worm spins on the bed and silk fiber is found on the bed. The silkworm appear creamy white as they are full of silk. The worms shrunk in their length. If all these maturity signs are exhibited by a silkworms, then we have to transfer them from rearing bed to the mountages. Process of mounting. The mature silkworms are picked from the rearing bed and transferred to the mountages. During mounting, care should be taken to put the right number of worms on the mountages to avoid overcrowding and formation of the double cocoons. Diseased worms of rearing beds should be picked separately and discarded either by burning or burying. Mounting density. 90 to 100 larvae should be mounted per square foot. Paper should be spread on the floor below the mountages to observe urine from the worm and change this paper when they become damp. Environmental condition during mounting. Temperature and humidity, these two factors affect the quality of the cocoons. Therefore, during mounting, temperature should not exceed 26 degrees Celsius and humidity should be in between 60 to 70%. While spinning, there should be minimal disturbance to the silkworm larvae. Now we are going to discuss regarding types of cocoonages or mountages. The most important device that supports the silkworm larvae for comfortable spinning of their cocoon is known as cocoonage or mountage. Cocoonage or mountages support the silkworm larvae for comfortable spinning of their cocoon. Mountage determines both quality and quantity of the cocoons. Different types of mountages are used in different parts of the India. In general, mountages are made up of wood, bamboo, cardboard, plastic, dry leaves, etc. However, an ideal mountage should fulfill the following requirements. A mountage must be have convenient space with suitable dimension for spinning a proper size cocoon. A mountage should not allow formation of double or malformed cocoons. A mountage should have provision for cleaning the excreta of the spinning larvae. A mountage should be suitable for easy harvesting of the cocoon. A mountage should be cheap, durable, and easy to handle. In India, following types of mountages are generally used. That is 1. Chandrika, screen type mountage, plastic mountage, Japanese low cost rotary mountage, bamboo strip mountage, bottle brush mountage, plastic bottle brush mountage. Now we are going to discuss about Chandrika. See, these are the Chandrikas. Chandrika, it is round or rectangular in shape. Chandrika is made up of bamboo mat. See, this is the bamboo mat. On this bamboo mat, a bamboo tape of 4 to 5 centimeter wide is on this bamboo mat. A bamboo tape of 4 to 5 centimeter wide is wound in a spiral manner. See, like this. About 1000 worms can be mounted on this type of mountages. Chandrika is popular in South India and West Bengal. Cocoons pinned on Chandrika are of good quality. Next, we are going to discuss regarding screen type of mountages. Screen type of mountages are made up of bamboo or wood. In this type of mountages, longitudinal strips are used instead of spiral tape as in case of Chandrika. This mountages screen can be folded and stored. This type of mountages can be kept clean and more ventilated so good quality cocoon can be obtained. Screen type of mountages are more durable than the Chandrika. Plastic mountage. It is made up of plastic instead of bamboo. It is more durable and easy to clean. They are more costly than the Chandrika, but here cocoon produced in these mountages are not equal in size. Japanese low-cost rotary mountage. In this mountage, a wooden frame of 4 longitudinal rods, 1, 2, 3, 4. In this mountage, a wooden frame of 4 longitudinal rods is attached by means of cross spokes at two ends to a central axis. Each rod has number of pegs placed at equal distance.
See the pegs. These pegs are connected by long threads of twisted rice straw in a rectangular pattern like that of charpoy. This montage is cheap, more durable, and make less chances of disease spread. Wattle brush montage. It consists of a thick coconut or jute fiber. The worm spin their cocoons in the space present between the leaves or in between the branches. This montages is very cheap and can be made easily as compared to the chandriki. Plastic bottle brush montages. It was developed by Central Sericulture Research and Training Institute, Mysore. It is a plastic rope with branches. Their surface is granulated in order to provide grip to the worms for spinning. These montages are durable and can be reused after disinfection. Paddy straw montage. It is a heap of paddy straw. Paddy straw montage is traditional one. It is very cheap, effective, and gives good yields. These are the disadvantages associated with the paddy straw montages. Here, cocoon may be crushed and harvesting is a problem. Harvesting of the cocoons. In silkworm, cocoons are harvested manually or mechanically by using harvesters. Cocoons are harvested on the 7th to 8th day from the inception of spinning. By this time, the worm will have completed spinning of cocoon and transferred into pupa. To confirm harvesting, cut 2 or 3 cocoons, check whether pupa are fully formed or not. The optimum time for harvesting is when the pupa turn brown in color and become hard in texture. Precautions during harvesting. Harvesting should be done after pupation is completed. Harvested cocoons should be spread in a single layer and not in heap to avoid heat generation. Early and late harvest of the cocoon should be avoided. Deflossing. After harvesting, all loose fiber on cocoon surface is removed, giving it a clean look and make ready for the market. Failure to deflossing of the cocoon lowers the marketability and price. Thank you.
Good morning, my dear friends. In this session, we are going to discuss about uses of silk and its byproducts. Silk, it is a most important natural fiber and used most commonly in the textiles. Silk, it is very soft, smooth and lustrous. All these qualities make the silk queen of the textile. Because of its very good strength, it is also known as biosteel. Now we are going to see different uses of silk. Silk is sold and exported as raw silk yarn. This is the raw silk yarn. Silk
woven into the fabrics and sold directly as fabrics or as ready-made garments. See, these are the ready-made garments which are made from the silk. Articles made from the silk like ties, scarves, soles, shawls, furnishings, carpets, and knitted materials, etc., have high market demand and high market value also. Silk yarn, which is obtained from tree molters, is used as package material in pencil industry and for making talcum powder puffs. These are the talcum powder puffs. Sound-free gaze are preferred by using silk as the raw material, and it is exclusively important in making precision machinery. In France, 22 to 24 denier silk is used in tire manufacture. The ties which are manufactured from this 22 to 24 denier silk has very longer lifespan than the ordinary rubber ties. Parachute is made from 13 to 15 denier silk fiber, and it was used during the World War II or Second, that helped in the tremendous progress of sericulture throughout the world. Cozy and soft sky jackets, comforters, and sleeping bags are also made from the silks. The silk gut is used in surgery for internal suturing. As it is a protein, it is auto-absorbable and need not to be removed after wound healing. Silk grafts used successfully to replace the cut arteries. Silkworms are extensively used in physiology researches to test different toxicants. Now we are going to discuss byproduct of sericulture. Sericulture involves four different activities. They are moriculture, large-scale egg production, rearing up silkworms, and reeling up cocoons. In each of these activities, a number of byproducts, popularly known as vestas, are generated. All these byproducts can be put to good use. Hence, it is claimed that in sericulture, nothing is a waste. Now we are going to discuss one by one byproducts of moriculture, byproducts of grainage operation, byproducts of silkworm rearing, and byproducts of reeling. First, we are going to discuss about byproducts of moriculture. If for some reason silkworm rearing is not carried out, in that case, mulberry leaves can be harvested, shade dried, and can be used for incorporation into the semi-synthetic artificial diet for the silkworm. Mulberry leaves are medicinally most important one. Excess of harvested leaves can be used as cattle feed, manure, or mulches for the mulberry garden. The rejected plant and its pods are used as the raw material for Paper pulp industry also used in furniture making and also used as fuel. Now we are going to see byproducts of grainage operation. Piercer cocoons, unused moths, and dead moths are the waste products which are obtained from grainage operations. Perforated cocoons are used to made gaicha and kachia and also used to produce spun silk in the mills. Unused moths, dead moths, and discarded eggs are dumped into the pit and allowed to form composts. Silk moth are used to brew medicinal wines, and this wine is used to treat impotence, abnormal menstruation, and menopausal symptoms. Byproducts of silkworm breeding: excess of harvested leaves, unfed leaves, and exuviae. All these are byproducts of silkworm breedings. All this waste can be used as compost and added to the soil. Rejected larvae, dead larvae, and cast larval skin can be used as poultry feed. Mature silk glands can be used as a source of guts which are used for surgical suturing. Silkworm litter can be effectively used as a raw material in the biogas plant along with the cow dung to produce fuel. Pharmaceutical and perfumery compounds are produced from the silkworm litter in China. Medicine developed from the chlorophyll, which is extracted from the silkworm excrement or excreta, is used as medicine for hepatitis and leukemia. Byproducts of reeling industry includes defective cocoons, pupal waste, and reeling wastes. Pupal waste can be used as compost. Pupa are used as human food as it is a rich source of protein. Pupa also used as animal feed and cattle feed. Oil is extracted from the pupa and it is used for burning the lamps and for preparing homemade soaps. Thank you.
Good morning, my dear friends. In this session, we are going to discuss about economics of silk production. India is the second largest producer of the silk in the world. First largest producer is the China. Indian sericulture industry providing employment to the 9.76 million people in rural and semi-urban areas. In India, the sericulture activities spread across 52,360 villages. India producing mainly four types of silks. They are Mulberry silk, Eri silk, Tazar silk, and Muga silk. Among all this silk, share of Mulberry silk is the highest one, that is 71%. Share of Tazar silk is 8%, share of Eri silk is 21%, and Muga silk is just 1%. The total silk production in India during 2021 and 2022 year was 34,903 million ton. It is an increase of 3.4% over the previous year. That is only 33,770 million ton. The major silk producing states in the country are Andhra Pradesh, Assam, Bihar, Gujarat, Jammu and Kashmir, Karnataka, Chhattisgarh, Maharashtra, Tamil Nadu, Uttar Pradesh and West Bengal. Among all these states, Karnataka is the leading producer with the contribution of 32% of the total silk production. This was followed by Andhra Pradesh with the contribution of 25% of the total silk production. The silk industry is one of the largest foreign exchange earner to the country. During the year 2021 and 2022, India's silk and silk product export valued 248.56 million US dollar. Export destination to the India. India's silk and silk products are highly demanded throughout the world. USA is the topmost importer of Indian silk and silk product. UAE is the second largest importer of Indian silk after the USA, followed by China, UK and Australia are the importer of Indian silks. See here country-wise share of Indian silk exporter given during the year 2020 and 2021. Government initiatives for the development and growth of the silk industry in India. The Government of India has implementing several research and development, trainings, transfer of technologies and IT initiatives. In addition to this, there are various schemes such as sericulture development in northeastern states, tribal subplans, silk samagra, and scheduled caste subplans are implementing by the government of the India for the development of the sericulture industry. The Indian Silk Export Promotion Council is an apex body of the exporters, manufacturers, and merchandisers. The main activities of this council are to explore the market, establish contact with potential buyers, organize buyer-seller meets, and organize silk fairs and exhibitions, resolve the trade disputes, promote and develop the Indian silk industry and exports. Thank you.
Good morning, my dear students. In this lecture, we are going to discuss regarding rearing equipment and appliances used in sericulture. They are rearing stand, rearing trays, and wells, chopping board, chopping knife, feathers, chopsticks, leaf preservation chambers, mountages or capunages, paraffin papers, foam rubber strips, leaf basket, and mats, etc. Now we are going to discuss all these equipments and appliances one by one in coming slides. See, these are the rearing stands. Rearing stands are made up of wood or bamboo and which are portable. These are the frames at which or over which rearing trays are kept. A rearing stand should be 2.5 meter in height, 1.5 meter long and 1 meter wide, and should have 10 cells. The trays are arranged on the shelves and each stand can accommodate around 10 rearing trays. These are the rearing trays. See, this is rectangular traditional rearing tray. This is rectangular plastic rearing tray. Rearing trays are used for rearing silkworm and are placed on rearing stands. There are many kinds of trays which are differing from one another in material, shape and size. Traditionally, rearing trays are made up of bamboo or wood so that they are light in weight and easy to handle. These are either rounded or rectangular in shape. Nowadays, rectangular plastic rearing trays are widely used in rearing of silkworms. Next, ant wells. Ant wells are used to stop ants from crawling onto the trays, as ants are serious threat to the silkworm. They are made up of concrete or stone. The legs of rearing stands rest on the center of the well and which is filled with the water. These rearing trays prevent the ants and other crawling insects. Next appliances is chopping board. Chopping board, it is made up of soft wood used for cutting the leaves of mulberry for feeding the silkworms. The size of the chopping board is 0.9 meter into 0.9 meter and 8 centimeter thick. Chopping knife. Chopping knife used for cutting the mulberry leaves. It is generally have a blade of length 50 centimeter long and 3.8 centimeter broad, and which is provided with a strong wooden handle. Feathers. Large white bird feathers are important for rearing silkworms. These are used for brushing the newly hatched worm to prevent injuries to them. Feathers also helpful while changing the bed or cleaning the bed. Chopsticks. These are tapering bamboo rods of size 17 to 20 centimeter long, which are thin, tapering to the end. These are used for handling and picking younger stages of the larvae to ensure the hygienic condition and to prevent damage to them. It helps to prevent contamination of disease among silkworms. Leaf preservation chamber. Leaf preservation chamber, these are used for storing the harvested leaves. The side wall and bottom are made up of wooden strips. The leaf chamber should contain a good number of ventilators covered with wet gunny cloths. Leaf chamber help in maintaining the leaf quality. Otherwise, leaves should be scattered on the floor in a single layer so that the water droplet on the surface of the leaf will be evaporated. A leaf should never be preserved in heaps. Mountages or cocoonages are the devices which support the silkworm for comfortable spinning of the silk. Mountages are generally made by using materials such as bamboo, plastic, dry straw, rotary cord, and coconut broomsticks, etc. Traditionally, mountages are made up of using large rectangular bamboo mats. But nowadays, plastic mountages, also known as netrikas, are widely used as they are light in weight and available at low cost. Cocoon from plastic mountages can be easily removed with dedicated cocoon harvester machine in a quick time, so no requirement of extra labors. Paraffin papers. These papers are used to maintain the required humidity in the rearing trays. These papers avoid the evaporation of moisture and keep the rearing trays humid. Foam rubber strips. To maintain the required humidity, thick foam pads soaked in water and are placed all around the beds. Leaf basket. Bamboo baskets are used for keeping and transportation of collected mulberry leaves from field to rearing places. Mats. Clean mats are placed under chopping board before chopping the leaves to collect the freshly collected leaves. Thank you.
Good morning, my dear friends. In this session, we are going to discuss about insect pest of mulberry silkworm. The insect pest of mulberry silkworm includes insects. They are parasitoids, predators, mites, pediculoides ventricoccus, vertebrates, and nematodes. Before going into the discussion about silkworm insect pest, we are here discussing some of the important terminologies. What do you mean by parasite? See here, Varroa mite is attacking honeybee. So, parasite is an organism which lives in or on the host body and obtains the food without killing its host. Then what do you mean by parasitoid? See here, Trichogramma wasp is a parasitoid. Uji fly is a parasitoid. So, parasitoid is a parasitic insect that lives in or on the host body and eventually kills its host. Parasitoids. It is a special kind of parasite in which immature stage is parasitizing and adult stage is free living. See here, this adult Uji fly just lay eggs. The maggots which emerge from the Uji fly's eggs or take care of killing up silkworm larvae. So, immature stage is parasitizing here and adult stage is free living. What do you mean by endoparasite? Endoparasites are the parasites they live inside the host organisms. Ectoparasites are the parasites which lives on the surface of the host body.
Then what is predator? See here, predating mantid. Predator is an animal which feeds upon the other animals which are generally smaller and weaker than them. Now we are going to discuss about the important insect pest of silkworm larvae, that is Uji fly. Uji fly's scientific name is Exorista bombycis. It is belong to the family Tachinidae and order Diptera. See, this is the Uji fly, and here Uji fly infesting silkworm larvae. The Uji fly is a serious endoparasite of silkworm which cause 10 to 15% loss to the silkworm crop to the tune of about rupees 400 crores to the Indian silkworm industry annually. See here life cycle of Uzi fly. It will be completed in 1 month and life cycle of silkworm completed in 2 months. Nature of damage of Uzi fly. Uji fly generally prefers third, fourth and early fifth instar larvae for oviposition or infestation. These Uji fly do not prefer the larvae which are mounted on mountages. Uji flies lays creamy white colored eggs in the intersegmental region of the silkworm larvae. From this egg, maggot will hatch out and this maggot bore into the silkworm body. After boring the silkworm body, this maggot feed on the tissues and internal content of the silkworm larvae. After 5 to 8 days, the mature maggots pierces the integument of the silkworm larvae and fall on the ground for pupation. If after Ujifly's infestation, silkworm larvae spawns the cocoon, in that case, the maggot comes out by piercing through one end of the cocoon and making the cocoon unfit for commercial reeling or for seed purpose. What are the symptoms or scenes due to Ujifly infestation or attack? We can see black spot on the surface of the silkworm larvae, creamy white oval shaped egg on the surface of the silkworm body, and perforated cocoons we can see generally. Management of the Uji fly. Uji fly is managed by adapting the IPM practices. They are cultural method, mechanical method, physical method, chemical method, and biological method. Cultural method includes collection and destruction of Uji fly affected silkworm larvae and cocoons. Destruction of Uji fly maggots and pupae. Mechanical method includes covering the individual rearing trays with nylon net, enclosing the entire rearing stand by using the nylon net, fixing the wire mesh to the windows and doors, and providing anti-rooms at the entrance of rearing house can exclude the Uji fly from infestation. Physical method includes keeping the Uji trap solution near the doors and windows, and this Uji trap traps this Uji fly. Chemical method include spraying 2% bleaching powder solution, dusting with levigated china clay, and spraying or dusting the ovicides or ujicides which kills the uji eggs. Here the table shows age of silkworm larvae and quantity of ujicide to be applied in different instars. Biological control method includes release of pupal parasite, that is Nizolinchus thymus, in the rearing rooms or the place where the mountages are kept. Legislative control includes avoiding transportation of Ujji-infested cocoon from infested areas to the new areas. When the above mentioned method uses individually, do not keep the pest incident below the 5%. Hence, integrated pest management package is developed for the suppression of this pest. Now we are going to discuss about second important pest of sericulture or silkworm. Dermestid beetle. Its scientific name is Dermestis cadaverinus or Dermestis eater. Adult beetle is black in color, oval in shape. Grubs are also black in color and covered with bristles or hairs. The grub causes more damage than the adult. This pest prevails throughout the year. What are the factors responsible for the pest outbreak? Storage of large quantity of moth-pierced as well as stifled cocoon over a long period of time responsible for the pest outbreak. Symptom of attack of this pest. We can see the presence of multiple irregular holes on the cocoons. This pest not only feed on the leftover or dead matter in the cocoon, they also attack the green cocoon as well as egg-laying moths whenever their population get increased. Dermestis beetle management involves two steps. They are preventive measures and control measures. In preventive measures, avoid storage of rejected cocoons and perished eggs. Keep the cocoon storage room clean. Drainage premises should be kept clean and tidy. Provide wire mesh to the door and windows to avoid free movement of the beetles and grubs. Control measures includes mechanical control, that is collection and destruction of grubs and adults of Dermestis beetle. Physical method: pack beetle infested cocoon in HDPE bags and expose to the sunlight for a period of 6 hours. Chemical method: wooden article of the cocoon storage room and drainage should be dipped in 0.2% malathion solution, then wash in water, sun dry, and then only reuse. Store the pierced cocoon in deltamethrin treated bags. Spray deltamethrin solution on the walls and floors of pierced storage cocoon room. Sprinkle commercial grade bleaching powder in the pierced cocoon storage rooms. Fumigate the dried cocoon storage room with methyl bromide. Can manage this pest. Ants. Ants are the pests which generally attack the silkworm larvae which are present in rearing trays and which kept over the rearing stand. So this pest can be easily managed by keeping the legs of the rearing stands in ant wells. Mites. Might see here images for the mites. Its scientific name is Pediculoides ventricoccus. Generally, female attacks silkworm larvae, pupae, and adults, take nutrition from them, and gives out a toxic substance which kills the silkworm. The body surface of the silkworm, which is struck with the mite, develop black specks.
The infested one lose appetite, become inactive, and have difficulty in excretion. And the excreta is attached to the bead-like to the anus. In case of severe infestation, the worm vomit yellowish green fluid. Control of this mite. Avoid the storage of wheat or rice straws near the rearing houses. Treat the building and touched material with acaricide or fumigate before use. Nematode. See here nematode. Its scientific name is Hexamermis microamphides. Nematode generally attack silkworms which are rearing in late autumn season. The nematode attacks young stage of the larvae and penetrate into their bodies. The head of the affected silkworms become transparent and the body milky white. Thank you. Good morning, my dear friends. In this session, we are going to discuss about important objective questions from Fundamentals of Entomology course. The scientific study of insect is the entomology. Study of external feature of an insect is known as insect morphology. Study of internal feature of an insect is known as insect anatomy. Any organism in which body is divided into head, thorax, and abdomen and possesses two pairs of wings and three pairs of legs is known as insect. Any organism which is noxious, destructive, or troublesome to the man or his interest is known as pest. Arthropods are originated during which period of Paleozoic era? Arthropods are originated during Cambrian period of Paleozoic era. Insects originated during which period of Paleozoic era? Insects are originated during Devonian period of Paleozoic era. The earliest known fossil insect is Raniella precursor. The insect came into the existence on the surface of the earth 300 to 500 million years ago. First edition of Systema Nature book published by Carl Linnaeus in the year 1738. 10th edition of Systema Nature book published by Carl Linnaeus in the year 1758. The ICZN code contains preamble and how many number of articles and glossary. The ICZN code contains preamble, 90 number of articles and glossary. A special account on termites of Tanjore district is published by J.C. J.G. Koenig. Father of taxonomy is Carl Linnaeus. First imperial entomologist from India is Hem Singh Pruthi. Indian Museum at Calcutta established in the year 1875.
ICAR established in the year 1929. Largest kingdom is Animalia. Largest phylum of animal kingdom is Arthropoda. Largest class of phylum Arthropoda is Insecta. Largest insect order of class Insecta is Coleoptera. Largest insect family is Curculionidae of the Coleopteran order. Silent Spring is an environmental science book authored by Rachel Carson. The textbook of agricultural entomology is authored by Hemsing Fruti. Some South Indian insect book authored by T.B. Fletcher. Indian insect pest and Indian insect life books are written by Professor Lefroy. In insect, exchange of gases take place through tubular structures are known as trachea. Tracheal opening on the body wall is known as spiracle. Holophenastic tracheal system seen in grasshopper, cockroach and dragonfly. Hypophenastic tracheal system present in headlouse and birdlice. Hyperphenastic tracheal system seen in columbola. Periodical process of shedding the old cuticle accompanied by the formation of new cuticle is known as molting. The cuticular part discarded during each molting is known as exuvia. The time interval between two subsequent molting is known as stadium. The form assumed by the insect in any stadium is known as instar. Molting in insect is controlled by endocrine gland, that is prothoracic gland. Commercial cultivation of BT in India started in the year 2002. Forest Research Institute located at Dehradun. DPPQS Directorate of Plant Protection and Quarantine Storage is located at Haryana, Faridabad. National Institute for Plant Health Management is located at Hyderabad. Spider belong to the class Arachnida. Number of segment in insect head are 6. Number of segment in insect thorax are 3. Number of segment in insect abdomen are 9 to 11. Insects' wings are located on meso and metathorax. Insect legs are located on pro, meso and metathorax ventrally. Eyes are located on ocular segment or first segment of insect head. Furcula or furca located on the ventral side of which abdominal segment in columbolan insect, that is fourth abdominal segment. Caudal breathing tube is present in water scorpion. Caudal filaments present in caudal gills are present in damselflies needs. Alimentary canal in insect is divided into anterior foregut, middle midgut, and posterior hindgut. In insects, alimentary canal actual digestion take place in the region of midgut. In alimentary canal, temporary storage of food take place in the region of crop. The cells of the hindgut harbors microorganisms are known as mycetocytes. Group of mycetocytes are known as mycotomes. Three layer of insect body wall are cuticle, epidermis, and basement membrane. Three layer of cuticle are epicuticle, exocuticle, and endocuticle. Chitin is absent in which layer of the cuticle? In epicuticle, chitin is absent. Female genital organs are located on the ventral side of 8th and 9th abdominal segment. Male genital organ located on the ventral side of 9th abdominal segment. Generally, spiracles are located 2 pairs on thorax and 8 pairs on the abdomen. In grasshopper tympanum, an auditory organ is located on the lateral side of first abdominal segment. 5 segments of insect legs are coxa, trochanter, femur, tibia, and tarsus. Longest and strongest segment of insect leg are is longest and strongest segment of insect leg is tibia. Longest and stoutest segment of insect leg is femur. Smallest segment of insect leg is trochanter. Antennae are also known as feelers. Antennae are located on antennal or second segment of insect head. Three types of antennal segments are scape, pedicel, and flagellum. Johnston organs are located on antennal, pedicel, or second segment. Types of mouthpot in housefly: sponging type of mouthpot in cockroach, biting and chewing type of mouthpot in plant bug, piercing and sucking type. Insecticide recommended for sucking pest are systemic insecticides. Types of mouthpot in honeybees, chewing and lapping type. Different organ of excretion in insect are Malpighian tubule, body wall, trachea, nephrocytes, etc. Major organ of excretion in insect are Malpighian tubules. Insect excrete uric acid. Most of the aquatic organisms excrete ammonia. Malpighian tubules are discovered by Marcello Malpighi. Post-embryonic change in the form of insect. Post-embryonic change in the form of insect during its life cycle is known as metamorphosis. A metamorphosis occurs in apterygote insect orders. Post-embryonic complete change in the form of insect is known as holometamorphosis. Post-embryonic little change in the form of insect is known as hemimetamorphosis. Post-embryonic no change in the form of insect is known as ametamorphosis. Polypod larva example caterpillar. Oligopod larva example grub of rhinoceros beetle. Apod larva maggot of housefly. Uptake pupa moth pupa. Chrysalis butterfly pupa. Tumbler mosquito pupa. Wriggler mosquito larva. Filiform antennae present in grasshopper. Moniliform antennae present in termites. Aristate antennae present in housefly. Serrate antennae present in pulse beetle. Geniculate antennae present in honeybees, pharygeal legs present in honeybees, posorial legs present in mole cricket, cursorial legs present in cockroach and ants, natatorial legs present in water beetles, sticking leg present in houseflies. Expand the following terms: DIPA, Destructive Insects and Pests Act, IINRC, IINRG, this is G, Indian Institute of Natural Resins and Gums, DPPQS, Directorate of Plant Protection, Quarantine and Storage, NIPHM, National Institute of Plant Health Management, AGMARC, Agricultural Produce Grading and Marketing Act, IARI, Indian Agricultural Research Institute, ICAR, Indian Council of Agricultural Research, LWO, Locust Warning Organization, FSSAI, Food Safety and Standards Authority of India, CIBRC, Central Insecticide Board and Registration Committee, NCIPM, National Center for Integrated Pest Management. Thank you.
Good morning, my dear friends. In this session, we are going to discuss about important objective questions from Fundamentals of Entomology course. The first question is: the dorsal sclerate of insect body segment is known as tergum or notum. The ventral sclerate of insect body segment is known as sternum. The lateral sclerate of insect body segment is known as the pleura. Moniliform type of antenna are found in termites. Filiform type of antennae are found in grasshopper. The type of metamorphosis seen in blister beetle is the hypermetamorphosis. Intermediate metamorphosis seen in scale insects, whiteflies and thrips. Alteration of generation in insect reproduction seen in aphids. Mushroom glands seen in male reproductive system of cockroaches. The neuron carry impulses from sense organs to the central nervous system are known as afferent neuron or sensory neuron. The neurons carry impulses from central nervous system to the effector organs are known as efferent neuron or motor neurons. Insect bloods lack in which vitamin? That is K vitamin. Zoology convention dictates that the name of the superfamilies ends in Oidea. Name of the family ends in idea. Name of the subfamilies ends in ine. And name of the tribes end in ini. The occurrence of diapause during summer due to high temperature is known as eastvation. The occurrence of diapause during winter due to low temperature is known as hibernation. The short period in between diastole and systole is known as diastasis. The synaptic gap is approximately 100 angstrom. Insect antennae are innervated by which part of the brain? That is, deuterocerebrum. The protein present in the insect wing joint is resilin, is the elastic and rubber-like protein. The real suture of the insect head is post-occipital suture. The phenomenon of storage of urea or uric acid in the fat body cells is known as storage excretion. Study and use of insects in crime investigation is known as forensic entomology. Body region of insect is known as body region of insect are known as tagmata. That is three region or three tagmata: head tagmata, thorax tagmata, and abdomen tagmata. In insect, the grouping of body segments into a region is known as tagmosis. Malphizian tubules are located at the junction of mid and hindgut. In insect larvae, abdominal legs are also known as pseudolegs or prolegs. Asymmetrical or cone-like mouth parts are present in thrips. Juvenile hormone is produced by Carpora allata. In solid feeding insect, midgut is lined with which membrane? That is peritrophic membrane. The phenomenon where the immature insects gives birth to the young one is known as pedogenesis or neoteny. The number of pairs of odoriferous glands present in stink bugs are 4 pairs. Most beneficial order in class Insecta is Hymenoptera order. Pupa remain enclosed in the hardened larval cuticle is the quarked type of pupa. Chitin is absent in epicuticle. Filter chamber is the characteristic feature of sap-sucking insect. Adult diapause seen in mango nut weevil. Egg diapause seen in silkworm. Larval diapause seen in pink bollworm. The conventional storage and release organ for neurosecretory cell is corpora cardiaca. Juvenile hormone secreted by corpora allata. Molting hormone secreted by prothoracic gland. Amplexiform type of wing coupling apparatus present in butterfly. Hamuli type wing coupling apparatus present in honeybees. Indian Insect Pest book authored by Lefroy, Maxwell Lefroy. Pseudohalters type wings are present in housefly. Telenium word related with Diptera order. Metapenastic spiracles are seen in wriggler, that is mosquito larva. Prophenastic spiracles seen in tumbler, that is mosquito pupa. Fringed type wings are seen in thrips. Long and many segmented cerci seen in mayfly. Forceps-like cerci seen in earwig. Asymmetrical type cerci seen in male embed. Male embed. Sclerotized or forceps-like cerci seen in earwig. Some South Indian insects. Book written by T.B. Fletcher. A pair of tubular structure found on the dorsum of aphid abdomen are called as cornicles. Chitin is formed by N-acetylglucosamine polymers. Furcula present in Columbola order insects. Style present in Thysanura order insects. The first instar larvae of blister beetle are called as trionglines. Trionglines. Insects generally excrete uric acid. Sperm storage organ in male insects is known as spermatheca. Fruit flies belong to family Tephrididae. Siphoning type of mouthparts are seen in butterfly. Sponging type of mouthparts are seen in housefly. Mouthparts consisting of stylates in hemipteran plant bugs and dipteran mosquitoes. Irreversible gland osmotoria present in citrus butterfly. DPPQS, Directorate of Plant Protection and Quarantine Storage, established in the year 1946. In insects, accessory pulsatile organs related to circulatory system. In insect, ganglia is related to nervous system. Pseudo-ovipositor present in housefly and fruit flies. Needle-like ovipositor present in cricket. Scientist who published an account of black insect is J. Kerr. The book Systema Nature is written by Linnaeus. Halocuticular invagination of body wall are known as apodemes. Here solid solid cuticular invagination of body wall are known as apophysis. Elytra type wings seen in beetles. Tegmina type wings seen in cockroach, grasshopper. Hemielytra type wings seen in plant bugs. Membranous type wings seen in bees, wasps, and dragonflies. Aristate type of antenna seen in housefly. Capitate type of antenna seen in butterfly. Geniculate type antenna seen in ants, honeybees. Fossorial type of legs seen in mole cricket. A running type of legs seen in cockroach ants. Jumping type of legs are seen in grasshopper. Opistognathus type of head seen in plant bugs. Prognathus type of head seen in beetles. Hypognathus type of head seen in cockroach, grasshopper. Cone type of mouth parts are seen in thrips. Expand DIPA, DIPA.
Destructive Insects and Pests Act IINRG IINRG Indian Institute of Natural Resins and Gums Indian Institute of Natural Resins and Gums DPPQS Directorate of Plant Protection Quarantine Storage Directorate of Plant Protection Quarantine and Storage NIPHM National Institute of Plant Health Management AGMARK Agricultural Produce Grading and Marketing Act IARI Indian Agricultural Research Institute ICAR Indian Council of Agricultural Research LWO Locust Warning Organization FSSAI Food Safety and Standards Authority of India CIBRC Central Insecticide Board and Registration Committee NCIPM National Center for Integrated Pest Management. Thank you.
Good morning, my dear friends. In this session, we are going to discuss about important objective questions from Fundamentals of Entomology course. Entomology is a which word? Entomon is insect, logos is study. Study of insect is the entomology. So entomology is a Greek word. Insects are originated during which period of Paleozoic era? Insects are originated during Devonian period of Paleozoic era. The insects came into existence on the earth 350 to 500 million years ago. 10th edition of Systema Nature book published in the year 1758. In India, Insecticide Act enforced in the year 1971. First imperial entomologist from India is Hem Singh Pruthi. Insect body wall is also known as integument or exoskeleton. Silent Spring is an environmental science book authored by Rachel Carson. In insects, exchange of gases take place through tubular structure are known as trachea. Cuticular ingrowth of body wall providing space for muscle attachment is known as endoskeleton. Endoskeleton is the cuticular ingrowth of body wall provides space for muscle attachment. In insect head. In insect head, epicranial suture is also known as line of weakness or ecdysial suture. Active immature stage between the egg and pupal stage of an insect is larvae. Chitin is formed by N-acetyl glucosamine polymers. to get protection against adverse conditions and natural enemies. The pupa is enclosed in a protective cover is known as cocoon. In sap-sucking insect, thin-walled bladder-like structure pass directly from anterior part of midgut to the hindgut without passing through the middle portion of the midgut, thus preventing the excess dilution of the hemolymph enzymes and facilitate better enzyme activity is known as filter chamber. Insects generally excrete uric acid. Sperm storage organ in male insects is Spermatheca. Fruit flies belong to the family Tephrididae. Siphoning type of mouthparts are seen in butterflies. Mouthparts consisting of stylates in plant bugs. Irascible gland osmotoria is present in citrus butterfly larvae. DPPQS, Directorate of Plant Protection, Quarantine and Storage is established in the year 1946. Hemulite type of wing coupling apparatus present in Hymenoptera. In insects, accessory pulsatile organs related to circulatory system. Pseudo-ovipositor present in fruit flies and also in houseflies. Scientist who published an account of black insect is J. Kerr, which insect shows intermediate metamorphosis. Thrips. Hibernation in insect is caused by low temperature. The book Systema Nature, written by Linnaeus. Halocuticular invagination of body wall. Halocuticular invagination of body wall are known as apodemes. The number of pair of odoriferous gland is present in stink bugs. Stink bugs possess 4 pairs of odoriferous glands. Most beneficial order in class Insecta is Hymenoptera. Pupa remain enclosed in the hardened larval cuticle is known as coarctate pupa. Chitin is absent in epicuticle. Chitin is absent in epicuticle. The membrane that protects midgut epithelial cell is known as peritrophic membrane. Adult diapause is seen in nut weevil, that is mango nut weevil. The dorsal sclerate of insect body segment is known as notum or tergum. Moniliform type of antenna are seen in termites. The type of metamorphosis seen in blister beetle is hypermetamorphosis. Alteration of generations in insect reproduction seen in aphids. Mushroom glands seen in cockroach. The neuron carries impulse from sense organs to the central nervous system are known as afferent neuron or sensory neuron. Insect blood lacks in vitamin K. Superfamily ends with the suffix oidea. The juvenile hormone is secreted by corpora allata. Elytra type of wing is present in beetles. Tegmina type of wing present in cockroach. Hemielytra type of wing present in red cotton bug. Membranous wings present in dragonflies. Aristate antenna seen in houseflies. Capitate antenna seen in butterflies. Geniculate antenna seen in ants. Fossorial legs are seen in mole cricket. Hamuli type of wing coupling apparatus is present in honeybee. Cone type of mouth is present in thrips. ICZN, International Commission on Zoological Nomenclature. International Commission on Zoological Nomenclature. BT, Bacillus thuringiensis. GEAC, Genetic Engineering Approval Committee. Genetic Engineering Approval Committee. BIS, Bureau of Indian Standards Act. Bureau of Indian Standard Act. DDT, dichloro diphenyl trichloroethane. Dichloro diphenyl trichloroethane. Thank you. Good morning, my dear friends. In this session, we are going to discuss about important objective questions from Fundamentals of Entomology course. Name the second segment of insect antenna. Pedicel. The major morphological structure useful in class insecta classification is wings. Insect head is made up of 6 number of segments. Major excretory product in insect is uric acid. The occurrence of diapause during winter due to low temperature is known as hibernation. Cuticular part discarded during insect molting is known as exuvia. The synaptic gap is approximately 100 angstrom. Study and use of insects in crime investigation is known as forensic entomology. In insect, the grouping of body segments into a region is known as tagmosis. Malphizian tubules are located at the junction of midgut and hindgut. In insect larvae, abdominal legs are also known as prolegs or pseudolegs. Cone or asymmetrical type of mouthparts present in thrips. Molting hormone produced by prothoracic gland. Peritrophic membrane in the insect midgut is the characteristic feature of solid feeding insects. What is a phenomenon where the immature insect gives birth to the young ones? Pedogenesis. Pedogenesis is a phenomenon where the immature insects gives birth to the young ones. DPPQS, Directorate of Plant Protection, Quarantine and Storage, at Faridabad, established in the year 1946. Insects belongs to the phylum Arthropoda. Spider belongs to the class Arachnida. Number of segments in insect thorax are 3. Insect wings are located on meso and metathorax. Anal cerci present in cockroach. Caudal gills present in damselflies needs. In insects, alimentary canal. Actual digestion take place in the region of midgut. Chitin absent in epicuticle. Male external genital organ located on the ventral side of ninth abdominal segment. First segment of insect leg is coxa. Johnston organ located on antennal pedicel segment. Major organ of excretion in insects are Malpighian tubules. Type of metamorphosis in Procura is anamorphosis. The number of pairs of odoriferous glands present in stink bugs. There are 4 pairs. Stink bugs have 4 pairs of odoriferous glands. Most beneficial order in class Insecta is Hymenoptera. Pupa remain enclosed in the hardened larval cuticle is known as coarctate pupa. Young one of dragonfly is needs. The membrane that protect midgut epithelium in insect is peritrophic membrane. Larval diapause seen in pink bollworm larvae. The ventral sclerate of insect body segment is known as sternum. Alteration of generations in insect reproduction seen in aphids. Mushroom glands seen in cockroach. The neuron carry impulses from central nervous system to the effector organ are known as motor neuron. Insect blood lacks in vitamin K. Family ends with the suffix idea. The conventional storage and release organ of neurosecretory cells is corpora cardiaca. Humuli type of wing coupling apparatus seen in honeybees. Indian insect pest book authored by H.M. Lefroy. Pseudohalters seen in Sepsiptera. Telenium is observed in Diptera. Metapanastic spiracles are seen in mosquito larvae or wriggler. Scaly wings are seen in butterfly. Long and many segmented cerci is present in mayfly. Prophenastic spiracles are seen in mosquito pupa or tumbler. Halters are seen in houseflies. Forceps-like cerci seen in earwigs. Dipa, Destructive Insects and Pests Act. IINRG, Indian Institute of Natural Resins and Gums. DPPQS, Directorate of Plant Protection, Quarantine and Storage. NIPHM, National Institute of Plant Health Management. AGMARC, Agricultural Produce Grading and Marketing Act. Thank you. Good morning, my dear friends. In this session, we are going to discuss about important objective questions from Fundamentals of Entomology course. Entomology, entomon is insect, logos is study, is a which word? It is a Greek word. Study of insect is the entomology. Insects are originated during which period of Paleozoic era? Insects are originated during Devonian period of Paleozoic era. The insects come into the existence on the earth 350 to 500 million years ago. Tenth edition of Systema Nature book, published in the year 1758. In India, Insecticide Act enforced in the year 1971. First imperial entomologist from India is Hemsing Pruthi. Insect body wall is also known as integument or exoskeleton. Silent Spring is an environmental science book authored by Rachel Carson. In insect, exchange of gases take place through tubular structure are known as trachea. Cuticular ingrowth of body wall providing space for muscle attachment is known as endoskeleton. In insect head, epicranial suture is also known as line of weakness or ecdysial suture. Active immature stage between the egg and pupal stage of an insect is known as larvae. Chitin is formed by N-acetylglucosamine polymers to get protection against adverse conditions and natural enemies. The pupa is enclosed in a protective cover is known as cocoon. In a shaft-sucking insect, thin-walled bladder-like structure pass directly from anterior part of midgut to the hindgut without passing through the middle portion of the midgut, thus preventing the excess dilution of the hemolymph enzymes and facilitates better enzyme activity is known as filter chamber. Insects generally excrete uric acid. Sperm storage organ in male insects, Spermatheca. Fruit flies belong to the family Tephrididae. Siphoning type of mouthparts are seen in butterflies. Mouthpart consisting of stylates in plant bugs. Irreversible glands, osmotoria present in citrus butterfly larvae. DPPQS, Directorate of Plant Protection and Quarantine Storage, established in the year 1946. Hemuli wing coupling present in Hemuli wing coupling apparatus present in Hymenoptera. In insect, accessory pulsatile organs related to circulatory system. It is not respiratory system, it is circulatory system. Pseudo-ovipositor is present in fruit flies and houseflies also. Scientist who published an account of black insect is J. Kerr. Which insect shows intermediate metamorphosis? Thrips. Hibernation in insect caused by low temperature. The book Systema Naturae written by Linnaeus. Hollow cuticular invagination of body wall are known as apodemes. The pair of odoriferous gland present in stink bugs. Stink bug has 4 pair of odoriferous glands. Most beneficial order in class Insecta. In class Insecta is Hymenoptera. Pupa remain enclosed in the hardened larval cuticle is known as coarctate pupa. Chitin is absent in epicuticle, the membrane that protects midgut epithelium in insect is peritrophic membrane. Adult diapause is seen in nut weevil, mango nut weevil. The dorsal sclerite of insect body segment is known as notum or tergum. Moniliform type of antennae are seen in termites. The type of metamorphosis seen in blister beetle is hypermetamorphosis. Alteration of generation in insect reproduction seen in aphids. Mushroom glands seen in cockroach. The neuron carry impulses from sense organ to the central nervous system are known as afferent or sensory neuron. Insect blood lack in vitamin K. Superfamily ends with the suffix "oidea". The juvenile hormone is secreted by corpora allata. Elytra type of wing is seen in beetles. Tegmina wing is seen in cockroach. Hemielytra type of wing is seen in red cotton bug. Membranous wing is present in dragonfly. Aristate antenna seen in houseflies. Capitate antenna present in butterfly. Geniculate antenna present in ants. Fossorial legs present in mole cricket. Hamulite type of wing coupling apparatus seen in honeybees. Cone type of mouth seen in thrips. ICZN International Commission on Zoological Nomenclature. BT Bacillus thuringiensis. ZEGEAC Genetic Engineering Approval Committee. BIS Bureau of Indian Standards Act. DDT dichloro diphenyl trichloroethane. Thank you.
Good morning, my dear friends. In this session, we are going to discuss about important objective questions from Management of Beneficial Insects course. The first question is, the weevil employed to kill water hyacinth is, that is, hydrophilic weevil Neochatina bruci. Bees are capable of flying at a speed of 25 to 30 km per hour. The comb of the damer bee is made up of a dark material is known as cerumen. The honey bee, which is cultivated in Newton beehive in India is, that is, Indian bee Apis cerana indica. The father of modern beekeeping in India is A.S. Atwal. The test insect used in the advanced genetic research is fruit fly Drosophila melanogaster. Indian Bee Journal is published by All India Beekeepers Association, Kolkata, West Bengal. The path
through which silk from China passed many countries and reached to the Europe is known as Silk Road. What is used to dust on molting worm for quick drying of the bed? That is lime powder. The number of kilograms of cocoon required to obtain 1 kg of reeled silk is known as rendita. Rendita. In female mulberry silkworm, sex glands present on the ventral side of 8th abdominal segment are known as Izivada glands. Lac collected before swarming is known as airy lac. The appendage lost during first molt are regained by which either male or female nymph during last molting in lac is, that is male lac, it regain it lost parts or lost appendages. Holoptic eyes are present in drone honeybees. Cooling of the colonies by the bees is done by fanning of the wings. The wild and cultivated flora, which are source of food material to the honeybees, are collectively known as bee pasturage or bee forage or bee flora. The fruit of mulberry plant is known as sorosis. The mulberry planting system adopted in Nilgiri area or slopy land is angular system of planting. World Bee Day is celebrated on every year on May 20. Which honeybee construct parallel comb? That is Indian bee and Italian bees construct parallel comb. The equipment made of a tin or copper container which is attached with a leather bellow used to subdue or calm down the bees while handling is known as smoker. The major constituent of honey is, that is fructose. The rearing house as well as the appliances used in rearing silkworm should be disinfected with 2% formalin. Major constituent of the silk filament is fibroin. The process of transferring the ripen silkworm larvae to the mountages is known as mounting. Paraffin vapor used in silkworm rearing to maintain humidity. To maintain humidity, paraffin vapors are used in silkworm rearing. Dermastid beetle attacks which life stages of silkworm? That is egg-laying moth as well as cocoon. Appearance of irregular dark brown spot or black spots on the body of silkworm larvae indicates symptoms of protozoan disease, that is pebrine disease. The sex differentiating gland in male silkworm larvae is Herold's gland, a small milky white body. Which state in India is the leading producer of mulberry silk? That is Karnataka. The place
where the disease-free layings of silkworms are produced is known as grainage. The flockery disease in silkworm is caused by bacteria. Largest producer of the lac in the world is India. In sericulture, one denier represents weight of 9,000-meter long silk thread. Pollination by butterfly is known as psychophily. The state which ranked first in lac production in India is Jharkhand. For chalky rearing, which leaf method is adopted to pick the correct leaves? That is glossy leaf method. Mother moth examination is done to detect which disease of silkworm, that is pebrine disease of silkworm. The celiac is processed into what? By heat process or solvent process, that is shellac. The celiac is processed into shellac by heat process or solvent process. Lac contain what percent of resin? Contains 68% of resin. The study of insects in relation to the crime investigation is known as forensic entomology. What is the characteristic feature of mulberry plants? That is the presence of idioblast, an enlarged epidermal cells in the leaf is the characteristic feature of mulberry plants. The word sericulture is derived from Greek word that is serikos. It means silk. The process of killing the pupae inside the cocoon to prevent the moth emergence is known as stifling. Golden yellow color silk is produced by Muga silkworm Antheria azama. The female pupa of mulberry silkworm is identified with X-shaped line or fine longitudinal line on the eighth abdominal segment. In sericulture, disinfection of rearing house and appliances is done by 2% formalin. Mulberry silkworm eggs changes to a color prior to 1 day of hatching. That stage is known as blue egg stage. Larvae of Eublemma amabilis and Holocochera pulveria are the predator on lac insect and lac also. The type of parasitism where many individuals of a species attack a single host is known as superparasitism. Expand the followings. IINRG, Indian Institute of Natural Resins and Gums. NBB, National Bee Board. TriFed, Tribal Cooperative Marketing Development Federation of India Limited. CBRTI, Central Bee Research and Training Institute. APEDA, Agricultural Products Export Development Authority. FSSAI, Food Safety and Standards Authority of India. KVIC, Khadi and Village Industries Commission. CSRTI, Central Sericulture Research and Training Institute. The substance extracted from diptera maggots
or fruit flies. Useful for wound healing is allantoin. Fertilizer schedule for mulberry irrigated row system is 300 is to 120 is to 120 NPK. The Tukra disease in mulberry garden is transmitted by means of pink mealybug, that is Macronellicoccus hirsutus. Hazel of white disease is caused by endoparasitic tracheal mite, that is Acarapis woodi. The book Bee and Beekeeping in India is written by Abrol. The enzyme which contained in the saliva acts upon sucrose of the nectar and convert it into dextrose and levulose is, that is invertase. National Bee Board is located at New Delhi. Basket type of traditional honey hives are known as scapes. The hive tool used to reduce the size of the brood chamber during the dearth period is dummy division board. The most ideal planting system for mulberry cultivation under rain-fed situation is pit system. Mulberry variety suitable for intercropping in coconut orchard is Sahana. Royal jelly contains a compound which exhibits antibiotic activity against bacteria and fungi is 10-hydroxydecanoic acid. The insect pollinator of oil palm is weevil, that is Elaedobius camerunicus. Elaedobius camerunicus. The pheromones secreted by the queen honeybee useful for maintaining colony cohesion and sterility of the worker bee is queen substance. Middle leg of the honeybees are modified for the purpose of picking wax plates. The resinous substance exuded by birds, leaves, and other parts of various trees and shrubs used for sealing cracks and crevices of honeycomb is propolis. Equipment which is used to prevent the movement of queen bees from brood chamber to the super chamber is known as queen excluder. This is the queen excluder. The beautiful caraman red dye, the beautiful caraman red dye is obtained from the dried and powdered body of which insect, that is cactus scale insect Dactylopius coccus. A method of reproduction in which a part of the colony migrates to a new site to make a new colony is known as swarming. The ideal soil pH for mulberry cultivation should be around 6.2 to 6.8. Scientific name of the wax beetle, the predator of the honeybee, is Platybolum alliviarum. The biofertilizer used for initiation of the root on the mulberry cutting is Azospirillum. Smallest honeybee is Apis florea. Largest honeybee is Apis dorsata. Lac insect belong to the family Caridae. Non-mulberry silkworm belong to the family Saturnidae. The most important insect used as bioindicator for the climate change is butterflies. The bee space for movable frame hive is proposed by L. Longstreth. The space required between any two frames for bees to move around is known as bee space. The honey yield of Apis cerana indica is 5 kg per hive per year. The mite used for the management of water hyacinth is Orthogalumna terebrantis. Orthogalumna terebrantis, which honeybee is called as a darling of beekeeping, that is Apis mellifera Italian honeybee. Honeybee sting is a modified form of ovipositor. Type of reproduction in lac insect is ovoviviparity. Royal jelly is secreted from which organ of the honeybees? That is pharyngeal glands. The honeybee larvae is fed continuously with royal jelly up to 8th day, and such cells are further packed with royal jelly and are sealed to develop queen honeybees. Aleppo gall is produced in oak trees. Royal jelly lacks vitamin E. Plant which supplies good quantity of pollen to the bees is sorghum. Plant which is good source of nectar to the bee is tamarind. Bees generally keep the hive temperature around 33 to 35 degrees Celsius. Honeybee possess how many pairs of wax glands? Totally 4 pairs of wax glands, that is on 3rd, 4th, 5th and 6th abdominal segments. Organisms which feed on dead and decaying organisms are known as scavengers. The queen bees leave the hive only during mating, flight and swarming. Which mulberry planting system accommodate more number of plants for unit area, that is triangular system? Suitable mulberry variety exclusively for late-age silkworm is G4. Trichogramma is a egg parasitoid. Easel of white in honeybee is caused by tracheal mite, that is Acarapis udi. Bald brood in honeybee is caused by lesser wax moth, that is Achoria grisella. An example for scavenger is chafer beetle. Lantana camara weed is managed by Ophiomia lantana fly. Cellular term related to egg laying. Few mulberry varieties are V1, G4, S1, S1 635, S146. Causal organism of Flaceri disease in silkworm is bacteria. Causal organism of Grassery disease in silkworm is virus. Major constituent of the silk is fibroin. Important pollinators are Bombus species, honeybee, sylphid flies. Honeybee chalkbrood disease causal organism's scientific name is Ascosphaera apis. Honeybee stoneroot disease causal organism's scientific name is Aspergillus flavus. Partenium weed managing bioagent is Zygogramma bicolorata. The most important insect used for advanced genetic studies is Drosophila melanogaster. Tukra disease in mulberry is caused by pink mealybug, that is Macronellicoccus hirsutus. Chandrika is a type of mountage. Block boxing of silk egg is done for uniform hatching. Prepupal parasitoid of sugarcane topshoot borer is Isotoma javensis. Thank you.
Good morning, my dear friends. In this session, we are going to discuss about important objective questions from Management of Beneficial Insect course. First question is, the comb of the dammer bee is made up of A dark material is known as cerumen. The test insect used in the advanced genetic research is fruit fly Drosophila melanogaster. Insects which feed upon the other harmful insects are known as entomophagous insects. Basket type of traditional honey hives are known as skeps. Bees are capable of flying at a speed of 25 to 30 kilometers per hour. The enclosed man-made structure in which honeybees live and raise their young ones are known as beehive. The plant which is a very the plant which is a very rich source of pollen to bees is sorghum. The plant which is a very rich source of nectar to bees is tamarind. The plant which is very rich source of pollen as well as nectar to bees is cotton. Name the equipment used for the protection of the head and face from the bee sting is bee wheel. The honeybee which is cultivated in Newton beehive is Indian honeybee, that is Apis cerana indica. The honeybee which is cultivated in Longstrath beehive is Italian bee Apis mellifera. The father of modern beekeeping in India is A.S. Atwal. Four legs of the honeybees are modified for the purpose of antennal cleaning. Equipment which is used to prevent the movement of queen bee from brood chamber to the super chamber is known as queen excluder. A method of reproduction in which a part of the colony migrates to a new place to make a new colony is known as swarming. The most ideal planting system for mulberry cultivation under rain-fed condition is pit system. The hive tool used to reduce the size of the brood chamber during the dearth period is, that is the dummy division board. The biofertilizer used for initiation of the roots on the mulberry cuttings is azospirillum. Mulberry variety suitable for the intercrop in coconut orchard is Sahana. Tukra disease in mulberry is caused by mealybug. The larva is fed continuously with royal jelly up to 8th day, and such cells are further packed with royal jelly and are sealed to develop a queen. queen is continuously fed with royal jelly. The bee space for mobile frame hive is proposed by L. Longstrath. Wax picking legs in honeybees are mid-legs. Mid-legs are wax picking leg. Darling of the beekeeping is Apis mellifera. Holoptic eyes are present in drones. Equipment made of a tin or copper container attached with a leather bellow used to subdue the bees or calm down the bees while handling is known as smoker. Honeybee sting is a modification of ovipositor. Cooling of the colonies by the bees is done by fanning the wings. In worker bee, royal jelly is secreted by pharyngeal gland or hypopharyngeal glands. The bald brood disease in honey bee colony is caused by lesser wax moth. The wild and cultivated flora, which are source of food material to the honeybees, are collectively known as bee pasturage or bee forage or bee flora. World Bee Day is celebrated on May 20. Type of mouthparts in honeybees is sieving and lapping type. The valuable material produced by certain gall insect is tannic acid. The ectoparasitic mite of the honeybees is Varroa jacobsoni. The study of insects in relation to the crime investigation is known as forensic entomology. The fruit of the mulberry plant is known as sorosis. Dammer bee or stingless bee belongs to the family Meliporidae. Replacement of a queen by another queen is known as supersedure. The mulberry planting system adapted in Nilgiri areas or slopy areas is angular system of planting. Pollen basket is also known as corbicula. Important pollinators are bombus species and honeybee. Bombus species and honeybees. Example for scavenger is suffer beetle. Axil of white disease is caused by mite, that is Acarapis woodi. Chalkbrood disease in bees caused by Ascos, Ascospira apis fungus. Ascospira apis fungus. Stonebrood disease in bees is caused by Aspergillus flavus fungus. Aspergillus flavus fungus. Biocontrol agent for mealybugs is Cryptolemus mantrugeri. Cryptolemus mantrugeri. Parthenium weed is managed by Zygogramma bicolorata beetle. Expand the followings. CBRTI, Central Bee Research and Training Institute, located at Pune. NBB, National Bee Board, it is located at New Delhi. KVIC, Khadi and Village Industries, Khadi and Village Industries Commission, EIC, Export Inspection Council, Export Inspection Council, TriFed, Tribal Cooperative Marketing Development, Tribal Cooperative Marketing Development Federation of India Limited, IINRG, Indian Institute of Natural Resins and Gums, APEDA, Agricultural Products Export Development Authority, Agricultural Products Export Development Authority, FSSAI, Food Safety and Standards Authority of India, CSRTI, Central Sericulture Research and Training Institute. Thank you.